Yep.
Good morning and welcome. I'm Charlie Wilson, the Director of UTSA Neurosciences Institute. Today is a special day for us. It's the day of our annual Neuroscience Symposium. Every year we set aside unusual responsibilities and get together for a wonderful day of total immersion in research topics selected by the audience. This year our symposium is organized by Dr. Lindsay Smith. And in accordance to our custom, Lindsay has selected a group of the best investigators in her field. Her teach us all about her topic, which is chemical senses and digestive system associated brain pathways. This is the 12th annual symposium in this series. Of course, this year is exceptional. All previous symposia have been held in person, including social events and walking on the river and having Mexican foods someplace, and all that stuff is not happening this time, which is too bad. And we can hold the symposium going back to that format sometime in the future. But on the other hand, this format allows us to deliver the symposium to a much larger audience. So whether you're watching on WebEx or on YouTube, please contribute your questions and comments to the chat, and questions will be asked to the speakers during the discussion. Thank you to Lindsay for arranging this great event for us. I'm really looking forward to it. As always, also thank you to Dr. Sam Kawashi, the Associate Director of the Institute, Symposium Mentor, and now also WebEx Guru. And you'll be seeing more from Dr. Karashi later. In addition to the WebEx event, the symposium is being streamed on our YouTube channel, UTSA Neuroscience. Thank you to Stuart Wilson, and yet another miracle the internet is producing our YouTube stream in today's story. And thanks very much to all the speakers who are taking the time to participate, and everybody watching and asking questions. So, Lindsay, let's go ahead and get started.
Thanks very much, Charlie. It's indeed my pleasure to bring together this wonderful panel of scientists today to delve into the fascinating field of chemosensory signaling and the roles of the peripheral chemosensory circuits that inform and modulate our behavior. I think it's great timing that Thanksgiving is only 2 weeks from today, the biggest eating holiday of the year for Americans. I hope these talks will give you more insight and appreciation of the molecules, cells, and circuits that contribute to the enjoyment of meals. So my goal for this symposium is to bring together leaders in this exciting field to highlight many of the processes that are involved in detecting chemicals within our food as it is digested, and how these signals can be relayed and modulated to influence behaviors. Our symposium will begin with Dr. Ellen Spector from Florida State University, giving an overview of chemical signaling in the alimentary tract, starting with taste.
Wrong.
My talk will follow Alan's with a focus on taste circuit wiring and rewiring in the tongue and gut. After a break for lunch, Dr. Ivan de Araujo from Mount Sinai School of Medicine will present his investigation of the vagus nerve and the physiology of reward. Dr. Diego Bojorquez and Dr. Maya Kelber from Duke University School of Medicine will then describe their exciting work on neuropod cells and fast synaptic signaling from the gut to the vagus. Dr. Suzanne Appleyard from Washington State University will enlighten us as to how signals from the vagal ganglia can be modified by brainstem circuits. And Dr. Lucy Shear from USC will discuss how both oral and post-oral sensors can contribute to glucose appetite. So as Charlie mentioned, we are on this WebEx format or on YouTube live stream. Please remember, if you have any questions, contribute the questions to the Q&A panel, and we will monitor them and bring them up during our Q&A sessions after each talk. So it's very much my pleasure to introduce our first speaker, Dr. Ellen Spector. Dr. Spector majored in psychology at Penn State University. He received his PhD in psychology at Florida State University, where he worked with James Smith to investigate radiation-induced taste aversion. And then he went on to do a postdoc at UPenn with Harvey Brill, where he was extremely productive, contributing to many important studies on the pathways for physician taste aversion learning. He started his independent career as assistant professor at the University of Florida, where he rose to the Florida to Florida State, where he's now Distinguished Research Professor of Psychology and Neuroscience. The work in Dr. Spector's laboratory focuses on the neural basis of gestation, using animal psychophysics, looking at the sensory processes and regulation of ingestive behaviors, as well as taste preferences and aversion. I was really thrilled when Dr. Spector agreed to participate in the symposium today, not only because I admire his work greatly, but because he has successfully bridged what I believe is really an artificial divide between taste chemosensation and the study of ingestive behaviors. Because of Dr. Spector's work and the work of all of our panelists speaking today, I believe that gap is really narrowing significantly. Please join me in welcoming Dr. Spector, who will give us his perspectives on chemical sensing as an entry to the alimentary tract and its multifaceted role. Alan, I believe you're muted.
Okay, let me get this set up. Okay, and hopefully you can see my slide. And so let me go there. And thanks so much for that very generous introduction, Lindsay. And I want to thank you for organizing this, and also give my thanks to UTSA Neuroscience Institute for inviting me to speak. And I'm really very excited to hear all the other talks in this very timely upgrade symposium. Now, as all of you know, the alimentary tract, let me know things it's just one long tube. Starts here, ends here, and as liquids and solids are placed in the mouth and swallowed, the contents engage a variety of sensory receptors that send signals to the brain and indeed also trigger action activities. And also, the nature of the stimulus changes as the food is physically and chemically processed for absorption and or excretion. And so what the mouth sees is not exactly necessarily the same stimulus that, say, the intestine sees. Now, the signals that the brain receives can be neural through the 5th, 7th, 9th, 10th cranial nerves, depending on where you are in this tube. Also, there's splanchnic afferents going through the spinal cord innervating the guts, and it can be humoral as well. And the neural pathways are often tertiary and carry somatic and or autonomic effects. And the brain integrates all of this sensory input, and this ultimately contributes to the control of ingestive behavior and metabolism. And today we'll be hearing a lot about how that occurs. Now, the concept of the gut-brain axis and neural integration of signals arising from the alimentary tract is not new. Depends on how far back you want to travel in time, but perhaps a reasonable starting point, especially with respect to neural integration, would be the post-World War II surge in brain research. And one of the pioneers study the controls of feeding, Elliot Speller, shown here, who was at the University of Pennsylvania. And this is a figure from Steller's classic tome in 1954 entitled Physiology of Motivation. Note all the arrows. Now, of course, the specifics and the complexity has grown over 3/4 of a century's worth of research. But the basic recognition that a variety of signals arising from different sensory systems are integrated by the brain in the service of this final common path of ingestive behavior, it's all there in 1954. Now, most of my professional life has been focused here, and I've been consumed by trying to understand the role of the gustatory system with respect to what role it plays in the symphony of signals arising from the alimentary tract. And I've been asked to give an overview of the gustatory system and then provide some examples from my own research program and the literature of how the functional organization of this critical sensory system can be interrogated behaviorally. Now, everything that someone eats or drinks has to first pass the scrutiny of the taste buds, which are distributed in distinct fields in these papillae in the oral cavity. And in this sense, they stand guard over the rest of the alimentary tract. So it goes without saying that the gustatory system is part and parcel of any kind of mechanism that is involved with the control of food. So let's start with the business end of the gustatory system, the taste bud. Taste buds are collections of about 50 to 100 specialized epithelial cells, many of which serve as sensory receptors. For the most part, the molecular receptors are expressed on the apical membranes of these cells, which protrude into a pore in the epithelium, allowing or exposing them to the oral cavity. Now, these cells form synapses, which relay the signal to the brain. And some of these synapses are referred to as unconventional, and they involve the release of ATP as the neurotransmitter through a calcium homeostasis modulator channel, where they bind with a purinergic receptor, specifically the P2X2X3 receptor found on afferent nerve terminals. Other types of synapses are more classical, especially those on what's called type 3 cells. Now, there have been at least 3 different types of cells that have been characterized in taste buds, type 1, type 2, type 3. They've been identified by the morphological characteristics and their expression of various markers. And this slide shows some of the transduction pathways for the conventional taste stimuli. And with respect to some of the key identified taste receptors, in rodents, we know that sodium-specific taste is mediated via the epithelial sodium channel, the ENaC, which incidentally is expressed in a variety of sodium-absorbing tissues, including the kidney and the colon. And the taste cell type that expresses these is somewhat unknown. It's a little bit controversial. It may be a type 1 cell. Acid taste, which is sour humans, is mediated primarily through the odopetrin 1 channel, recently identified by Emily Lyman's group, and these are expressed on type 3 cells. Now, sweeteners, both natural and artificial, bind with the T1R2 and T1R3 proteins that form a heterodimer, and these are expressed on a distinct subpopulation of type 2 cells. L-amino acids bind with the heterodimer formed by the T1R1 and T1R3 proteins, and these are expressed also on a distinct and segregated subpopulation of type 2 cells. And then finally, bitter ligands bind with one or more of the 30 or so T2R proteins that are expressed on yet a third distinct and segregated subpopulation of type 2 cells. Now, there are other proposed receptors and transduction mechanisms as well. These include those for non-sodium salts, glucose. Lindsay Shear later on in the day will be talking about that. Fats like CD36, GPR120, maltodextrin, calcium. And I don't have time to go over all these, nor do I have time to go over all the interesting features of the slide that many of my colleagues in the field work very hard to discern. But and some of these may soon be outdated as more information comes in. But I do want to make a few points. First, many of the receptors found in the oral cavity are also found in other tissues, especially in the gut. And a subset of taste bud cells appears to express only a single class of receptor, And this provides the basis of what's called a label line code. And by label line code, what that means is that activity in a receptor cell or neuron in the system is necessary and sufficient for the generation of a qualitative percept. However, there's other taste bud cells. There's some very nice new data from Catherine Medler and Amory Torregrosa, along with a history of papers that have found that some taste bud cells appear to respond to more than one perceptual class of stimuli. And this has been taken as evidence for an across-neuron pattern code or an ensemble code, where taste quality is coded across a pattern of activity in the population. Now, that sort of brings us to the question of, well, what happens at the ganglion cell level? And these are data from calcium imaging experiments conducted in Charles Zucker's lab, in which responses of the geniculate ganglion neurons, and I should say that the geniculate ganglion, those neurons innervate the taste buds of the anterior tongue and the palate. And they were looking at responses of these neurons to prototypical taste solutions that were placed in the oral cavity. And as you can see, the vast majority of these neurons responded to only one perceptual class of stimuli. However, they also identified some of these neurons that were multi-responsive. And honestly, these data actually resemble the outcomes of the elegant single fiber recording experiments conducted by Mary and Frank 40 years ago. Now, these are data from Steve Roper's group, and he also found that there were many narrowly tuned neurons in the geniculate ganglion. But he also found a class of broadly tuned neurons. And what was interesting is when he increased the concentration of the taste stimuli, this class seemed to expand. But there were still some narrowly tuned units. And the way that he characterized this is a metric very commonly used in the field called entropy. As far as from information theory, it ranges from 0 to 1. When the score is 0, it means the cell is completely narrowly tuned, and the score of 1 means that it's broadly tuned. And you can see when you increase the concentration, the average entropy value went up. And so this has been taken as evidence for an ensemble code, perhaps, and that may be true. But one must consider that these entropy values still on average are rather low, although admittedly some of these cells had high values. But perhaps more important is the label for the multi-responsive neurons may not be related to perceptual quality. It may be signaling some other characteristic or feature of stimulus, which brings me to what I think is a very important consideration. Taste means different things to different people, even specialists. And that's because the nature of the sense of taste is multidimensional and can be divided into at least 3 grades. So you have sensory discriminative behavior. This involves processes that lead to the identification of the amount and the quality of stimulus. But then there's this motivational or affective domain. And this refers to processes that promote or discourage ingestion. And then finally, there's this physiological domain, so-called cephalic phase reflexes that prepare an animal for the optimal digestion and assimilation of nutrients. And when one is interpreting behavioral or neurophysiological data, it's important to be mindful of this functional framework, because some methods assess function in one domain, but not another, and some assess function in both or all 3. And so I'll come back to this framework more explicitly shortly. Now, this is a schematic of the ascending anatomy of the gustatory system in rodents. Rodents are the most common animal model of taste function for experimental research. Believe it or not, it's oversimplified, as my colleagues will attest. But it basically shows the pathway that taste signals take as they travel from the different fields of taste buds in the periphery on their way through to the higher regions of the brain. And I want to point out a couple things. First, these four different nerve branches innervate different fields of taste buds in the oral cavity, and they project in a rough orotopic fashion to a region of the caudal brainstem known as the nucleus of the solitary tract. And then second, you'll see that signals from the viscera, especially the gut, travel through the vagus nerve and terminate right behind the taste area in the caudal nucleus of the solitary tract. And in fact, in general, as you trace the taste pathway through the brain, the visceral pathway is always right next door. And when one considers the complete alimentary tract, as I started off my talk showing, this makes complete anatomical and functional sense. Now, through nerve transection and regeneration experiments conducted in the rat, it appears that stimulus identification depends largely on the information from the gustatory branches of the seventh cranial nerve. It turns out that, at least to date, the signals in the glossopharyngeal nerve are unnecessary for stimulus identification, despite the fact that it innervates by far the most taste buds. However, it's the input of all three of these nerve branches that contribute more or less to the affective and motivational aspects of taste processing, depending on the stimulus. And the reason I point this out is because already in the periphery, there's evidence that you have some partial segregation of channeling of input to the brain on the basis of its function. And finally, there's plenty of evidence supporting the importance of the ventral forebrain taste nuclei being involved in taste-related motivational and affective processing. But the role of the thalamocortical pathway, sort of classical and niscal type pathway, in sensory discriminative processing remains controversial or at least unclear, but that's another talk for another day. So what I'd like to do for the rest of my time is to demonstrate how one can interrogate these different domains of taste function. And to do this, especially given the theme of the symposium, I'm going to provide some examples from gastric bypass research that we've been conducting in my lab along with my collaborators. And but first, I have to tell you a little bit about some of the features of gastric bypass. This is what a Roux-en-Y gastric bypass looks in the rat model that we've adopted. It's very similar to the major replumbing of the gut that occurs in humans. Ingested foods and fluids go straight through this gastric pouch right into the jejunum. The remnant stomach, the duodenum, and proximal jejunum is anastomosed distally into the jejunum so that bile and digestive enzymes can have access to the ingested contents. This is truly a remarkable surgery. I mean, there's a lot of research that's ongoing, both preclinical and clinical. Patients lose about 25% of their body weight, sometimes more, along with sleeve gastrectomy. This is one of the most effective treatments for weight loss. Weight loss is maintained in many patients for 20 years. All type 2 diabetes improves, and some patients go into a long-term partial remission. Hypertension improves, and overall, it's estimated there's a long-term survival benefit of approximately 30% risk reduction, and that's driven primarily by reduced cardiovascular events and reduced cancer. Now, if you would have asked me what bariatric surgery was 15 years ago, I wouldn't have been able to tell you much, but that's when I met my good friend and colleague, Dr. Carl LaRue, and that began over a decade's worth of experiments on the effects of gastric bypass in the rat, as well as humans. In my own lab, we're only doing rat model research, which blossomed into international collaboration collaborations over the years. I currently have an NIH grant with Carl and Barbara Livingston, who's at Ulster University. Their labs are conducting studies on patients with bypass, and my lab is conducting complementary experiments with our rat model. Now, all of these collaborations, including my current one, over the years have shared one theme, and that is to adapt and apply behavioral procedures for human testing that don't rely on verbal report and that can also be used in animal models. And we feel that this allows for a more direct assessment of target behaviors. Animal models can be used to approach underlying mechanisms, and ultimately we feel this enhances the translational relationship between human and animal model findings. Now, just like humans, after bypass, rats stabilize at a lower body weight, they eat less, and they have elevated levels of key gut hormones such as GLP-1 and PYY. Now, it turns out that under various testing conditions, rats will display substantially decreased intake and preferences for sugar solutions relative to water. And a similar decrease in preference is seen with fat emulsions as well. But I'm going to focus on sugar today. And this is in seeming agreement with some of the changes in preference for high sugar, high fat foods that are reported by patients with bypass. And importantly, in rats, this decreased preference never drops below indifference, okay? So it's not like they're completely avoiding the solution. By the way, these data were collected by Dr. Marco Buder, who, when he was a fellow in Carl's lab, he's now chief of bariatric surgery at the hospital of the University of Zurich. And importantly, Marco showed that not only did this not drop below indifference, but it's specific for sugars and fats, so that preference aversion functions for sodium chloride, quinine, citric acid were unaffected by gastric bypass in rats. So there's a lot of different factors that can come into play when you're measuring preference and choice in feeding or drinking over a long-term period like 24 hours. But is it possible that the surgery is affecting sensory discriminative aspects of taste function? Patients with bypass often report changes in taste sensations, especially with respect to sugar. So how would one test for this? Well, one way to test for this is to simply measure psychophysical thresholds. And in humans, some studies have found increased sensitivity, that would be decreased thresholds, after gastric bypass, but other studies have found no effect. And in animal models, it just hasn't been tested. And how would you test this in an animal model? Well, we make use of a specialized behavioral testing device called a gustometer. And this has several methodological features that make it well suited for testing sensory discriminative taste function. First, it allows us to deliver small volumes of taste stimuli and to measure immediate responses. And these two features increase the confidence that the behavior of the rat or the animal under examination is under or a sensory control. Second, it allows us to use taste stimuli as a cue for other reinforcing events. And thus, the affective properties of the stimulus itself aren't really driving the responsiveness. It's just simply serving as a cue. And so the workhorse procedure that we use in the lab is something we refer to as the two-response taste discrimination procedure. We can train animals to discriminate taste from water and then decrease the concentration of the taste. To measure detection thresholds, we can train animals to discriminate one taste from another taste. Generally, the animals are water deprived, which motivates them to sample a small amount from a spinning ball in this gustometer. And they're given 5 seconds to make a choice. If it was taste, they're trained to lick a right tube. It's actually a ball, and If it's water, the train to lick the left tube would counterbalance those sides across the animals. If the response is correct, then they get reinforced with water, which is reinforcing because they're thirsty. If it's incorrect, they get a timeout, just like you do with your kids. And then there's an intertrial interval when we clean the drinking spout and dry it and return it back for the next trial, clean and uncontaminated. Let me show you an animal working in this task. The animal sampling from the spinning ball behind that slot. And I believe this was the water side, so it got it correct. It was water. And now the sample ball's being cleaned. And when the light comes on, the animal will be available, or the spout will be available for the animal to sample. It's taking a bath now. Okay, sampling. And it's a taste, I think. And I thought it was water, but he got it correct. It was a taste, and he got his reinforcer. And this is how we clean the drinking ball, and then there's some air that blow dries it. And now the animal is sampling again, and here's one of the response balls, and water comes out of the bottom. Now, training and testing takes many weeks. This is really kind of a grueling procedure. We have test days interposed between stimulus control days. Those stimulus control days, we present clearly detectable concentrations to make sure that the animals still know what the rules are. And at the end of testing, however, okay, all this effort goes into it, but we're able to plot psychometric functions for each animal that provide several parameters of performance shown on this graph, an asymptote slope, and the most important of which is probably this EC50, which provides us with the location of this curve, and in essence serves as our operationally defined detection threshold. Now, my graduate student, Kelly Hyde, for her dissertation, used this type of procedure and assessed the effects of high-fat diet versus lab chow maintenance on sodium chloride and sucrose taste detection thresholds in female rats. Women are the majority of patients who receive bariatric surgery. And she also looked at the effects of gastric bypass or changing the high-fat diet to a lab chow diet. And this is showing the weight loss that's very characteristic after bypass surgery. Now, I'm not going to go into all the details of this experiment. I don't have time, but I'm going to cut right to the chase. These are, whoops. Oh, yep. Here we go. These are the sodium chloride detection thresholds, and I can sum this up by saying that there were no main effects of diet. There was no there were no main effects of gastric bypass surgery. There was, however, a diet by gastric bypass interaction, and that was driven primarily by the fact that the bypass animals on the high-fat diet group had about a 4/10 of a log unit lower threshold in sodium chloride, and that means they were more sensitive than the sham-operated controls. We weren't really expecting this, and we're still trying to wrap our heads around the specific outcome. Kelly was making these slides last week. She just finished testing, and she's doing other tests now. We're a little concerned about this one outlier, whether that's driving this up. But nevertheless, in terms of cross-sham groups in general, and the other groups of gastric bypass, there didn't seem to be any effect. And more importantly, with the focus on sugar, there were no effects of any of these manipulations: high-fat diet maintenance, gastric bypass, on sucrose thresholds. And the failure to find an effect of high-fat diet on these thresholds is interesting, because there's evidence that maintenance on a high-fat diet has detrimental neurobiological effects on the gustatory and olfactory systems. For example, in the case of the gustatory system, There's evidence from Robin Dando's lab at Cornell that maintenance on a high-fat diet decreases the number of taste buds. So Kelly, at the end of this experiment, will be taking oral tissue samples from her rats, and she plans to examine the anterior and posterior tongue taste buds. And with respect to the failure of gastric bypass to affect sucrose detection thresholds, it suggests that the changes in sucrose intake and preference are not due to a change in the strength of the signal with respect to sensory discriminative taste processing. Now, one caveat, of course, is that this task assesses only one aspect of sensory discriminative taste function, and it's not like we've tested every possible compound. However, I will say this with respect to sucrose. This is an assay that is highly sensitive to manipulations of the gustatory system, including nerve transection and nerve regeneration, brain lesions, and interference with taste receptors. Now, and I should also note that in this business, you need to demonstrate that the animal is under stimulus control and not responding to extraneous cues, which is every psychophysicist's nightmare. And so, a large piece of evidence for that, of course, is that animals still display concentration-dependent performance. But just to confirm that, we always fill the reservoirs at the end of these experiments with water and arbitrarily assign some as the taste stimulus and some as the water stimulus, and then we see if the rats can perform above chance. And as you can see, none of Kelly's rats performed above chance in this type of manipulation. Now, the task I just described is well suited for measuring the function of sensory, or in the sensory discriminative domain, because taste is merely serving as a cue to drive the choice, the reinforcements coming from the water reward when a proper choice is made. However, when we're interested in measuring function in the affective or motivational domain, we assess how well the taste stimulus itself drives responsiveness to obtain and or ingest it, or avoid it and reject it. And there's a variety of behavioral assays that we've used to assess taste function in this motivational affective domain, but today I want to focus on some new unpublished data using one of these procedures called the taste reactivity paradigm, which was developed by Brill and Norgren over 40 years ago and further advanced by the research of Kent Berridge. Now, this paradigm involves the measurement of stereotypical oromotor somatic behaviors referred to as taste reactivity. And these are in response to small volumes of taste stimuli delivered directly into the oral cavity through cannulae that are chronically implanted. And these behaviors are video recorded, they're coded, and they're scored blind. It's a very tedious process. You have to really need or want to do it. But when you do that, Brill and Norgren characterized two classes of responses. The first was referred to as ingestive responses. These are elicited by normally preferred stimuli such as sucrose in proportion to their concentration. They involve tongue protrusions, lateral tongue protrusions, mouth movements. I'll show you an example. Compliments of Dr. Frederick Setterholm. Oh, I gotta turn my laser pointer off. I hope you'll be able to see this. These are tongue protrusions and mouth movements. You'll see some tongue protrusions in slow motion. You can see the tongue is crossing the plane of the incisor. And somebody is counting all of these up in a like 30 or 60 second session. Now, the yin-yang of the brain, there's also the aversive responses, and these are elicited by normally avoided stimuli, such as quinine, in proportion to their concentration. They include gapes, chin rubs, head shakes, forelimb flails. Oftentimes, it's accompanied by fluid ejection all over the chamber. I'll show you a clip of a hallmark aversive response to gape. The corners of the mouth retract, exposing the lower incisors. I mean, it sort of looks like a gagging response. So my graduate student, Ginger Blondie, let me get my shown here, tested taste reactivity in male and female rats that had sham or bypass surgery. And the way she did this was by giving rats what's called an intraoral intake test and measuring how much the animals consumed until they rejected the. This is a paradigm that was also developed in the Grill Lab and further refined by the work of Bill Flynn and especially Joel Kaplan. And what Ginger also did was she filmed the first 30 seconds of the infusion and scored it on the water day and scored it on the first sucrose day and the last sucrose day. Now, as you can see, all of the groups, so she had males and females, and these groups had gastric bypass. These groups had sham surgery. These groups had gastric bypass. And you can see that on the first sucrose day, all the groups increased their intake. But you can also see that the gastric bypass rats, not unexpectedly, were drinking far less sucrose in this intraoral intake test. But what about their taste reactivity? I should mention the rats were non-deprived throughout testing, except we removed food 45 minutes before the test. Now, this is what happened on the water day. You can see there's some moderate ingestive behavior. This is the ingestive score. There's also some moderate aversive or a small amount of aversive behavior, especially in this male sham group for whatever reason, but not much, okay? And this is a very typical water response. But then on the first day of sucrose, you see there's absolutely no aversive behavior, and there is a substantial and significant, statistically significant increase in ingestive behavior. And not only that, but this is also shown that the pattern of ingestive behavior has changed. It's now with sucrose dominated more by tongue protrusions than by mouth movements, which is the light blue. And tongue protrusions, through a series of other experiments conducted by a variety of labs, including my own, seem to be a higher level of valence in terms of palatability, right? And so it's interesting that that pattern changes. And then on the last day, when Ginger tested them, you can see it emulates what happened on the first sucrose infusion day, and no aversive behavior and very substantial TP-dominated ingestive behavior. And so that means that after 7 sessions of sucrose infusion, there wasn't any development of what you would call a conditioned taste aversion, especially in the gastric bypass rats. But what was really fascinating was that Ginger noticed that immediately after the intraoral meal and the pump was turned off and the animal had passively dripped the stimulus, the bypass animals over sessions progressively started to display aversive taste reactivity, even though the infusion had stopped. And they didn't display it on the first session, so obviously experience is involved with the development of this response. But they showed it, across sessions in an increasing fashion. This is on the 8th sucrose infusion after the infusion had stopped. And you may be saying, oh, well, these are pretty low scores relative to the ingestive scores, but part of the reason for that is these aversive behaviors, they simply take longer to emit. So one thing that Ginger did was to score that one session in terms of the amount of time during the 30-second period where animals were displaying aversive responding. And you can see in the bypass groups, they were showing aversive behavior about half of the time or more on average, whereas the sham-operated groups were displaying virtually zero. And so we think that the development of this aversive taste reactivity after 8 sessions of sucrose infusions reflects some type of negative visceral state. And we don't know exactly what that state is. We have some ideas and we'd like to pursue some future experiments. But whatever this negative state is, it does not change the animal's initial palatability evaluation of the stimulus, at least as measured by taste reactivity, at the start of these future sucrose sessions. Let me summarize some of the implications of the results from these bypass experiments that I've described. So neither prolonged maintenance on a high-fat diet nor Roux-en-Y gastric bypass has any impact on sensory discriminative responsiveness to sucrose in rats, at least as measured by psychometric detectability curves. And in contrast to the prevailing view, the motivational and affective potency of the taste of sugar, and same thing with fat stimuli, although I didn't show you any data, does not appear to decrease after gastric bypass. And we've tested this in a variety of ways with other types of assays, and it always leads to the same conclusion, at least in our hands. And what we think is going on is that rats are adjusting their intake of sugar and fat to minimize the negative visceral consequences of their ingestion, and it likely involves some learning processes. And again, we're still conducting other experiments to pursue this further. Now finally, how can the physiological domain of taste function be investigated? This domain refers to the so-called cephalic phase responses. Ivan Pavlov won the Nobel Prize in 1904 for studying this, which he referred to as digestive reflexes. And by the way, this was several years before his work on conditioned responses. And a nice example of the taste-triggered cephalic phase response in rats can be found by a 35-year-old paper from Harvey Grill's lab, in which they infused small volumes of glucose and other sweeteners on other sessions into an oral cannula, and found that it was only the glucose that stimulated a pre-absorptive insulin release within minutes of its infusion without any observable rise in plasma glucose levels. Now, this figure only shows the results for glucose, sucrose, and maltose, but they also tried a variety of non-caloric sweeteners as well. John Glendinnings's group has recently replicated this phenomena in mice. And one of the differences in John's hand was it wasn't just glucose, but it turned out that other glucose-containing compounds, including sucrose, had some efficacy. And this is thought to be due to the presence of disaccharidases that Bob Margolski's group at Monell have found expressed on the apical membrane of a subset of taste receptor cells that are thought to break down disaccharidases, providing a source of glucose. And also you can see, interestingly, in this figure, that the response is independent of the canonical T1R sugar receptor, because T1R3 knockout mice are just as responsive as wild type to the glucose and sucrose, and they're not responsive, neither the knockout nor the B6 wild type mice are responsive to fructose, even though fructose binds with the T1R sugar receptor. Now, my colleagues, I'm a little disappointed. I was hoping to show you some of our own data. My colleagues Wolfgang Langens, Alan Watts, and Merta Arnold just started a grant to study cephalic phase responses to sugars in a rat preparation that's fitted with an intraoral cannula, as well as a gastric catheter and a hepatic portal vein catheter. The latter allows for very sensitive and temporally precise measurements of glucose absorption, as well as insulin release. And unfortunately, the COVID pandemic has just temporarily stalled this research program. So I hope I've accomplished my goal to provide an overview of the gustatory system and to convince you that taste function is multidimensional, and you have to consider that with respect to interpreting the many different neurobiological and behavioral findings that are produced. I also hope that you can see how one can experimentally approach the different domains of taste function, especially behavioral. And while at the same time providing some insight on the effects of gastric bypass on taste function in our rat model. And, this is a very simplistic slide here, and it's just making the point that these various domains of taste function, they don't operate in a vacuum. They can be influenced by not just gustatory input, but they can be influenced by chemosensory signals from other sensory systems. And really, this emulates the thinking of the pioneers of the study of the neural controls of ingestive behavior over 3/4 of a century ago. And I think I'll end there to give you some time for questions and answers. But before I do, I want to point out that really I did none of this. I would be lost without all the graduate students and postdocs and research faculty and technicians that I've had the good fortune to work with over the years, and of course my collaborators. These are the ones that have been involved with some of the data that I've shown you today, and of course there's a host of others. And also, I want to give a shout out to the National Institutes of Health. I'm indebted to the NIH for their support of my research program for many years. And I think I'll just end with that. So now we're going.
Thanks. Thanks, Alan. We're going to field some questions. So I think first I want to just let the attendees know that you do not have control over your unmute. So if you would like to be unmuted, you'll have to send me a little message in the chat. And then everyone who poses a question in the Q&A box, I will unmute you to ask your question personally. But first I'd like to offer up room to the panelists to ask questions. And if not, that's while I figure out how to unmute people in the audience. Okay. We do have a couple of questions in the Q&A. Maybe we can start with those. So I have one here from Rowan Calkins. Rowan, would you like to be unmuted? I'm going to unmute you, but he's asking about the animal models, what animal models were used. Rowan, you're unmuted if you want to ask your question.
Hi, thank you for unmuting me. Can you guys hear me?
Yes.
Okay, awesome. Glad to know that I can be the test subject for that. Good talk. This is a little bit out of my wheelhouse looking at taste, but it's super interesting to see. I had a question about mainly the tests where you were doing sucrose and the either gustatory or aversive behavior, but in general, I guess this could be applied. What line of rats are you using, or is it multiple?
No, yeah, I'm sorry. There's so many, right? The devil's always in the details.
Yeah.
There's so many critical details I've left out, and and many of these are important. It's a great question. We're using Sprague-Dawley rats.
Okay.
And I grew up with Sprague-Dawley rats, and so once I got started with Sprague-Dawley rats, that's what my lab has been using. Now, when we're using rats, it's always Sprague-Dawley, but we've done a host of taste research in mice as well, and we've used various inbred strains of mice as well as genetic knockout models, and those have included B6 and mice and DBA / 2J mice and 129 mice, SWR mice, and then we've done a lot of work with some of the knockout strains that have been generously provided to us, at least the breeders from Charles Zucker.
Okay, all right, thank you.
You're welcome.
Okay, I'm going to go ahead and unmute you, Claudia Perez. You can pose your question personally. Are you there, Claudia? Okay, don't think she's there.
Okay.
Okay. Okay.
Alan, I have a question. So the finding after the high-fat diet stimulate over time is very interesting. That you saw no change in the sucrose preference or detection thresholds. And you're right. I think this is really interesting because a lot of papers recently have been discussing changes in the peripheral taste system due to high-fat diet. So I mean, I know you're following this up, but I just want to push you a little bit to hypothesize about where this disconnect might be happening.
Yeah, no, absolutely. I'll tell you what I think. So it's interesting, and I don't want to be overly critical, but in the taste field and a bit in the smell field, that it's a little different than other sensory systems. I think like other sensory systems like vision and audition, the work, the neurobiological work that was conducted was driven by the psychophysics, right? It's a little bit the opposite. The taste and smell feel the behavioral work's been driven in the other way. And so I think I think that neurobiology is exceptionally important and provides us the things we want to know. What are the brain mechanisms? But when you see changes in the brain or in the peripheral nervous system, you can't automatically assume that it's going to have a behavioral consequence unless you test for that. And when you're talking about behavior, there's many different types of behavior. So, for example, I mentioned these different domains of taste function. Well, it could be that if we test these high-fat dieting animals, they're going to show some decreased responsiveness in another domain of taste function, okay? And not necessarily the sensory discriminative domain. Or, it could be that the high-fat diet, is maybe affecting some other compounds that we haven't tested. I'll also say in terms of taste bud number, because we did like decades of research on the rat model with nerve transection, regeneration, and counting taste buds. And it really seems like once you get over a certain number of taste buds, at least for the rat, in some of these tasks, they do amazingly well, okay? So it could be that the kinds of tasks that we're using are just obviously not sensitive for the kinds of changes you might expect. So until there's more research, more behavioral research that's done, looking at different taste compounds, looking at different types of functions, it's going to be a hard thing to answer. I don't contest that there are these changes in the periphery, but we need to understand what they mean functionally.
Okay, thanks. Any other questions? We are way ahead of schedule. Okay. I think Claudia Perez is here. She just doesn't have her mic available. So she's asking me about the RYGB study, the rat, why it had preference for sucrose rather than sham, and why high fat. Maybe some thoughts on that. I think that's what she's asking.
I'm going to have to reinterpret the question because the gastric bypass animals don't have the preference for sucrose. They show decreased preference in a 2-bottle test for sucrose and for fat stimulant. But I think maybe your question is like, why not other taste compounds? Well, sugars and fats are, they have calories in them, right? And so, it could just, and I think this is exactly what it is. I don't think it's a it's a change in taste. I don't even think it's a change in true, taste preference or taste reward. I think it has more to do with learning how to ingest those tastes because of the post-ingested consequences. I don't think it's a taste aversion per se. I think it's more akin to what would be referred to as a conditioned avoidance. So let me borrow an example from Marcy Pelchat's paper like 30 years ago with Paul Rawson to distinguish between like conditioned aversion and conditioned avoidance. And Harvey Grill it's been a great talk for Harvey Grill. And this was really an interesting paper, sort of looking at what types of conditions lead to avoidance in a two-bottle preference test and lead to the generation of aversive responses. And the way that they sort of provided an example of this in humans is to look at someone who has a conditioned taste aversion. We all have them, right? We're frequently observed to distinctly flavored alcoholic beverages. You eat something, it's relatively novel, you get nauseous, perhaps vomit, and then, you don't touch that perhaps ever again unless you extinguish. Now, take another example of a person who doesn't eat shrimp, okay? So one person may have eaten shrimp and got taste aversion, got nauseous. Another person may have eaten shrimp and avoids it as well, but they didn't have vomiting and nausea. Instead, they had an allergic reaction, a shellfish allergy. So you've got two individuals that are now avoiding shrimp. They're not eating. One individual, the person that had the vomiting and so forth, they find the palatability of shrimp disgusting. That the taste of it is not palatable at all. But the individual that had the allergic reaction still finds, the taste of shellfish delicious. They would like to eat it, but they avoid it because of its post-ingestive consequences. And so I sort of think this is perhaps what's going on in gastric bypass rats and perhaps even in patients. I don't know if I answered your question because I reinterpreted it.
Well, Alan, following up on the gastric bypass portion. So when you did the pairing with sucrose, eventually you would get these the aversive type of facial representations, right? So what do you think sucrose just happened to be, the tastant you chose? Would this happen with any tastant you paired? Do you think there's potential differences between caloric and non-caloric sugars? You're only giving them a little bit, so there's no real.
Oh, no, we're giving them a lot. No, so okay. Let me explain. So great question. I understand it. So first of all, I don't think that it's a taste aversion in the sense that we never see at the beginning of the session. Even on session 8, they were showing very positive type responses, ingestive responses, no aversive responses. So it's something that doesn't occur until the gut is full, right? And we're not force feeding them in the sense that they're able to drip this whenever they want. And obviously, the gastric bypass animals drink less of it than the sham operated animals. So, we scored at first 30 seconds of what's called the intraoral intake test till they naturally terminate the session by passively dripping it. And so then we turn the pump off, and there's a few details I've left out because they're a little complicated, but I don't think they're critical for what I'm trying to convey. And that is that once the session's terminated, then there's no more infusion. There's probably still sucrose taste in the mouth. And I think in the end, that's going to turn out to be important because it could be that the sucrose taste and the full gut together are sort of serving as a compound conditioned stimulus. It's possible. With respect to other types of stimuli, I think those are great questions. In fact, we had all these experiments planned that maybe I would have been able to show you some of the data. I hate using COVID as an excuse all the time, but this is really true. You know, ginger, we're still going to do them, we got away, but ginger was going to like cut the concentration of the sucrose in half. We were going to try, you know, we wanted to look at osmolarity. Now you could get fancier, you know, and implant some type of gastric catheter in these animals. It might be a little tricky because of the bypass procedure, but at least theoretically, from an experimental standpoint, you know, that would be one way to separate that out. Of course, Yvonne's group, I'm sure we're going to hear a lot about those kinds of experiments later. So, you know, yeah, you're raising great questions, but we're really, we were surprised and kind of very fascinated by the evolution of these responses at the end of the session.
So we have 5 questions lined up now. So I'm going to go ahead. Shannon Landon actually has a question. I'm going to go ahead and unmute her. Do you have a mic, Shannon? Can you?
Hi, can you hear me?
Yes.
Wonderful. First off, Dr. Sparks, that was a wonderful talk. I really enjoyed it.
Thank you.
I had a question about, so you mentioned the label line versus code or combinatorial code.
Yeah.
I was wondering how extraoral sensory input can modulate that type of mechanism, that type of transduction mechanism.
Okay, so there's several aspects to your question. So the first is like, can input from extraoral taste buds influence the coding of what, you know, signals that are coming from the oral cavity, right, and affect that code? And I think in a general sense, yes. You know, that was sort of my last slide, in the sense that I think visceral signals could have an impact on the gustatory system, and there's evidence of this. Susan Travers and Joe Travers and J.P. Baird have some interesting data recording from the parabrachial nucleus taste cells, okay, in the rat, and finding that they change the response properties on the basis of whether sucrose is in the stomach or not. Also, Ralph Norgren has similar data. You know, so I think visceral signals can modulate the responsiveness of taste neurons. Okay, there's a lot more work that needs to be done. On the other hand, I think it's a fascinating question in the sense that, for me, you know, I've been interested in the taste system, and we've been arguing about label, line, and cross neuron pattern codes forever, and we're still arguing about it, you know, and it's a fascinating argument. You know, it's like there's data supporting both points of view, and probably in the end, and it probably won't be solved in my lifetime, but, you know, there'll be some kind of combination of both. But I really think what's going to play heavily into the final verdict is going to be like what's being coded, right? Now, the reason I think this is interesting with respect to this particular session is that, you know, you have the same problem with vagal afferents, right? I mean, and splanchniks, like, you know, how are the dimensions of these different signals, you know, the nutrients, the stretch, you know, all these different factors, how are they being coded, you know? And eventually, if you're going to have integration, all these signals have to be speaking the same neural language so they can integrate, you know? And so a lot of it will also depend on where in the system, I think, you are in terms of recording responsiveness to visceral and gustatory stimuli. I don't know if I answered your question, but
Thank you.
The questions are piling up. We have, if we don't get to your question, we're a little short on time, but we can go a few minutes over, I think. I'm going to try to either address them in the panel discussion later, or else we can, you know, we can have some private conversations with Dr. Spector later. But I'm going to unmute you, Anne Marie, if you want to ask your question.
Hi, Alan. Great talk.
hey, Anne Marie.
So I have a question about the body weight. So we are finding that high-fat diet alone is not necessarily driving changes in taste-driven behaviors, and we're using a different task, right? We're using pre-access taste testing, which also involves motivational factors. But the level of obesity seems to be really important in our measures in mice. Your gastric bypass rats are around 400 grams when they start, which is not super obese for a rat. I was wondering about Kelly's animals. I missed, are those animals considered obese? Have you looked at fat mass to confirm obesity?
Yeah, so Kelly, I didn't show these data. First of all, there's no definition of obesity in a rat.
Right.
And the second thing is that there's a big difference between body weight in males and females. And so, you know, Kelly, of course, included, these were just females, but those female rats, she just got the data literally like 2 days ago or something. She did an echo MRI. We have one now at FSU. And she found that the females that were on the high-fat diet had, and I just can't remember the numbers, but they had 4 or 5 times the fat mass that other groups did. And so, you know, from that standpoint, is that obese? I don't know what the definition of obesity is in a rat, but certainly they were showing much more fat mass. And those, sorry, I'm sorry, I'm misspeaking. Those weren't the females. Those were male rats. She did a separate experiment without bypassing male rats, but the high-fat diet animals definitely showed this increased fat mass. So it's a good question, and I will say it's an important point because you know, it's really important to distinguish between the effects of the diet, especially, you know, tasting a very highly palatable diet every single day, right? Versus the obesity. And Kelly, of course, will, you know, eventually be looking at all kinds of correlations between these measures to see if they predict anything.
Okay, so we do have a bunch of other questions, but I think we'll hold on to these. And I think I'm going to go ahead and let's see. I'm going to mute you, Alan. Dr. Spector, we'll see your questions. Everybody who posed questions in the Q&A. Just want to assure you of that. Okay, so Lindsay, our next talk is Lindsay McPherson. I'm going to go ahead and introduce her, but let's see. Okay. So it's my pleasure to introduce Lindsay. She's been an assistant professor here at UTSA since late 2017. She did her doctoral work in molecular biology at the Scripps Research Institute with Artem Paputian. There she discovered that the pungent taste of garlic and onions activates TRPC1A pain receptors, and then went ahead and figured out the specific molecular mechanism of that activation. That work resulted in prestigious papers, national recognition in the form of the Norton Galula Prize, and an NRSA training fellowship. So from there, she moved on to a postdoc to expand her work on taste chemosensation into circuit mapping in the lab of Charles Zucker at UCSD and then later Columbia. She recognized that her research interests required new tools, so she built them. So as you'll hear in her talk, she used her genetics background to adapt a novel fluorescence transsynaptic complementation technology from C. elegans into both Drosophila and mouse. That key innovation yielded more high-impact discoveries, first about temperature representation in Drosophila, and then later, by combining genetics, in vivo calcium imaging, and behavioral analysis, she identified the guidance molecules that signal the exquisite wiring specificity of bitter and sweet taste receptors to their target neurons. So Lindsay's ever-expanding tools and research interests have won her funding from HHMI, Brain Research Foundation, NIH, and a number of other sources. In her short time here, she's distinguished herself as a incredibly generous colleague and mentor. Today, she's going to talk about continuing her path of innovation, expansion, and discovery in her new lab by stepping into the gut and interoception. So welcome, Lindsay. Take it away.
Thanks, Alma. All right, let me get my screen to share properly here. Minimize this. Okay. All right, hopefully you're seeing my screen now. All right, thanks so much, Selma, for the very kind introduction. It's distinctly a pleasure to give a little bit of background about my investigations of the taste system, as well as my new interest in investigating chemosensory connectivity throughout the digestive system, getting a grasp of chemosensory connectivity in the tongue and gut. So thanks to Alan's wonderful introduction, I don't have to spend a whole lot of time on the background of taste, but just to give my favorite tidbits about the taste system and why, I got so interested in investigating this wonderful, simple sensory system. So the taste field uses 5 major taste qualities: sweet, bitter, salty, sour, and umami, to investigate our 5 senses. So these are the canonical types of foods that contain these, taste types, although when you're eating, most of the time you're getting many combinations of these tastes at the same time, in addition to the flavors of that are introduced by the olfactory system in combination with taste. So our 5 basic tastes give us information about the nutritional value and palatability of our foods, giving us information about the carbohydrate content, which gives us the sweet flavor, bitter flavors, which might indicate toxins or potential irritants, salts within the food that give us the nutritional sodium that we need to balance our salts within the body. Sour indicates the presence of acids, and umami indicates the presence of amino acids within our food, like the breakdown products of proteins. And as Alan really nicely explained, we use different taste receptors to identify these molecules within the foods that we eat. This includes the T1R2, T1R3 co-GPCRs for sweet taste, the bitter family of T2R receptors, which contains about 30 different GPCR molecules, ENaC channels, which are responsible for sodium taste, autopetrin, which Alan just described, was very recently identified, finally, after so much time of searching, for the acid sensor proton channel. And umami is kind of a cousin of sweet, where it uses both T1R1 and T1R3 heteromeric GPCR proteins to detect amino acids within the food. And again, as Alan mentioned, these taste qualities kind of fall into 2 categories, where you get attractive responses to things that are sweet, umami, or mildly salty, and you can elicit attractive behavioral responses like licking of the lips and ingesting solutions. And these reactions are similar across mammals. If you give a baby something that's sweet, they'll lick their lips and want more. Similarly, you get a reaction that's very similar to that with the rats. On the other hand, if you give an aversive taste, you get aversive reactions in the orofacial responses to sour, highly salty, or bitter substances. And so you can see that there's these very nicely discriminable reactions to different taste qualities. And much of this is reflex action. Although, as Alan mentioned, you can learn to be aversive to something that was previously pleasurable. So just some basic anatomy of the taste system, and I don't have to go into too much detail because, again, Alan gave a beautiful introduction to this. Let me just get my highlighter going. So in the front of the tongue, and this is our mouse model, we have the fungiform taste buds, which contain 50 to 100 taste receptor cells that are all commingled really closely together. These taste receptor cells are innervated by the afferent nerve fibers originating from the geniculate ganglion for the fungiform taste buds in the front of the tongue. These geniculate ganglion neurons have these long processes innervating the tongue, and then their central processes go on to innervate this brainstem region, the nucleus of the solitary tract. In my lab, when I was a postdoc, my colleague, Robert Barreto, developed a really nice assay where we could look directly at the activity of individual geniculate ganglion neurons using this in vivo geniculate ganglion calcium imaging prep. So using careful surgery to expose the tympanicula and expose the geniculate ganglion itself, we could look at the activity of these neurons using GCAMP calcium indicators. So the GCAMP is expressed either using a transgenic mouse or using an AAV virus to express these calcium indicator within the geniculate ganglion. We do the surgery and then are able to perfuse different tastants over the surface of the tongue while we image the activity of these cells over time. And as you can see in these panels on the right-hand side, this is one geniculate ganglion that I've kind of split into 4 different panels. And you can see the reactivity of individual neurons really brightly lighting up with GCAMP fluorescence as we perfuse sweet, bitter, salty, and sour stimuli over the surface of the tongue. If we look more carefully at individual cells by looking at different ROIs, we can plot out the response profiles of individual neurons within the ganglion. And this is an example of this. So if you look at one cell, this is a green cell highlighted here, it responds quite nicely and strongly to sweet stimuli, but to not much else. This one responds to bitter, but not much else. This one to salty, umami, and sour. And if we look at many ganglion neurons over many mice, we can get an overall picture of how these neurons tend to respond. And as Alan mentioned, the majority of neurons respond to a single tastant, showing some specificity here. These to sweet, these to salty, these to sour, and these to bitter. But also there is a small minority of ganglion neurons that respond to multiple taste types that are multitude. So as Alan mentioned, this gives us an idea of a precise labeled line type connectivity model where individual taste receptor cell types are innervated by dedicated populations of geniculate ganglion neurons, sour to sour, sweet to sweet, bitter to bitter, and so on. But because the taste receptor cells are not neurons, they're neuroepithelial cells, and they have a finite lifespan of about 10 to 14 days on average, this taste bud is really a dynamic structure, and things are turning over all the time. This creates a problem, or conundrum, where the taste neurons, the afferents, innervating the taste bud have to reconnect over and over again to new taste buds, to new taste receptor cells as they're being formed and maturing within the bud. But we know that our sense of taste remains somewhat static. We don't have wild fluctuations in our ability to detect or to discriminate tastes over the course of a few weeks or a month. So this process of rewiring has to be somewhat regulated in order to ensure that our taste fidelity is maintained. So we hypothesized that the taste receptor cells must express specific molecules that provide either instructive or permissive signals in order to establish the appropriate connections with gustatory neurons. To identify which molecules these might be, my collaborator Ho-Jun Lee and I in Charles Zipper's lab used a molecular profiling approach to look at the expression patterns within bitter and sweet taste receptor cells. To do this, we created 2 transgenic lines, one that labeled the bitter taste receptor cells with the green fluorescent protein and the sweet taste receptor cells in the blue fluorescent protein. We bred these 2 lines together to get double transgenic lines, and then enzymatically digested the taste epithelium and sorted these 2 populations by fluorescence activated cell sorting. These were in the days before single cell RNA sequencing was all the rage. So we did pooled samples where we took all of the bitter cells together and all of the sweet cells in the pool and sequenced their RNA expression profiles. If we look at the differences between the bitter taste receptor cells here plotted on the y-axis versus the sweet taste receptor cells on the x-axis, we could see potential molecules that were specifically expressed by one of those two populations. So first off, we looked for our control genes, which we know should be specifically expressed in one population or another. Here in green are the population of bitter taste receptor cells, the bitter taste receptors, the T2Rs, and you can see they're highly expressed on this side of the dividing line, expressed within the bitter TRC population. And on the other hand, T1R3 and T1R2, the co-GPCRs for sweet taste, are highly expressed on the bottom side of the sweet line. So we asked whether there might be some specific molecules that would be able to form or be able to connect the neuron fibers to the sweet or bitter cell population. So we looked for anything that might be conducive to this, like cell adhesion molecules, axon guidance molecules, something like this. And what we found were two different semaphorin genes, semaphorin 3A, which is specifically expressed and highly expressed within the bitter taste receptor cells, and semaphorin 7A, which was more specifically expressed within the sweet population. We were very excited by finding these semaphorins because semaphorins are a family of secreted and membrane-associated proteins, which are widely expressed in the nervous system, but also expressed in other tissues. We were very attracted to them because they're known to be involved in axon guidance, neural circuit assembly, and synaptic specificity. And so the SEMA3A gene was specifically expressed within the bitter population, and it is a secreted version of the semaphorin. Semaphorin 7A is specifically expressed in the sweet population of TRCs, and this one is GPI anchored to the membrane surface. So going back to our ganglion imaging assay, again, if we look at control mice, most of the neurons respond to only a single taste quality. Only a few of them could be considered multi-tuned. But if we take a mouse that's lacking the SEMA3A gene, we find a very different profile. Instead of the singly tuned type of response, we see many, many more neurons responding to multiple taste qualities at once, giving these neurons that respond to sweet, bitter, salty, umami, and sour all at the same time. And there's many more of them than there were in controls. By looking at these profiles very carefully, we came up with a hypothesis that was actually the bitter neurons that tended to be responding to more than one taste quality. And then the other neurons, some of them were more normal, especially some that were just singly tuned to sweet or to salty, for example. So this gave us a model of looking at semaphorin 3A as responsible for kind of connecting the bitter taste labeled line, potentially. And that the loss of SEMA3A produces geniculate ganglion neurons that are confused in their wiring, such that the bitter neurons connect also to sour, to sweet, and to salty cells, in addition to their normal targets, the bitter taste receptor cells. To really nail down if this is happening, what we did was create a genetic model where we ectopically expressed semaphorin 3A, the bitter semaphorin, within the sweet population of taste receptor cells using the T1R3 promoter. When we did this and I investigated the geniculate ganglion response profiles, we saw that there was this massive increase in the number of sweet and bitter co-responding neurons. So in this case, they were not just mistargeted randomly, they were retargeted to connect to sweet in addition to bitter. Similarly, for semaphorin 7A, which we found to be co-expressed within the T1R3 population of taste receptor cells, the sweet umami ones, we could cause a rewiring effect if we expressed the semaphorin 7A in the bitter taste receptor cell population, this time giving the same type of profile where sweet and bitter responses were seen in the same geniculate ganglion neurons. But in this case, we think that it was actually the sweet neurons mistakenly becoming connected up with the bitter taste receptor cells. We could do this again by placing the semaphorin 7A under the control of the PKD2L1 promoter, which is expressed specifically within sour taste receptor cell populations. And these cells would then show a sweet plus bitter response, these neurons normally connecting to their sweet target cells, but also being diverted over to receive input from sour responding taste receptor cells. So this experiment shows that there's a nice potential, molecular determination of the connectivity within the peripheral taste system. And the nice thing about semaphorins is that quite a bit of work has been done on them in other fields. And so there are already known receptors for these molecules, one of which for semaphorin 3A is neuropilin-1. And neuropilin-1 we found is very nicely expressed in a subset of geniculate ganglion neurons. And we created a neuropilin-1 free line to hopefully investigate this further. By crossing the neuropilin free with a channel rhodopsin reporter line, we're now able to look at what happens when you activate this population of geniculate ganglion neurons innervating the tongue. So we set up an optogenetic lick-triggered light stimulation protocol where, using a gustometer similar to the one that Alan Spector has been using, we can have the mouse lick a spout and then trigger light stimulation to activate these neural fibers that are innervating right at the front of the tongue in the pungiform papilla. So if we do this in a control mouse where we have it always likes to play my experimental before my control, but I wanted to show the control first. So, okay. This one, so these mice are water restricted for 24 hours, so they're relatively thirsty, and they know that there's a spout behind the shutter, and the laser light comes on, and in this control mouse, it continues to lick no matter that it's receiving light stimulation on the tongue. It doesn't care too much, and when the light's off, it licks just as much too. However, the glutamate that it contains the NRP1 pre-driving expression of channel rhodopsin within this population of geniculate ganglion neurons, we can see what happens when the laser light hits the tongue. The mouse immediately pulls away, doesn't like that. But when the light's off, it's still very thirsty and will lick readily from that spout. So one question we have is this response to the laser just an aversive response, or is this actually signaling to the mouse that it feels something bitter? And these are questions that rotation student Ryan Wood is now looking at using a more complicated behavior box assay where we have to train the mouse to go right and left to detect bitter or sour stimuli and ask whether or not the mouse found the stimulus to be more close to a bitter sensation or just generally aversive in one way or another. So in summary, we found that the semaphorin molecules play a very important role in promoting connectivity between taste receptor cells and gustatory neurons. That this is a special example of an chemoaffinity code with very good cellular specificity. And we believe that the semaphorin 3A neuropilin receptor ligand pair likely work together to help rewire bitter taste connectivity during taste receptor turnover. And we wonder whether the plexin 7A, semaphorin 7A, plexin 1 receptor ligand pair may do the same for sweet, but we haven't investigated this in detail yet. But these data tend to validate and uncover a mechanism for establishing and maintaining modality-specific connectivity in the peripheral gustatory system. But as Alan mentioned, there is that population, right, of multiple responding neurons and potentially multiple responding taste receptor cells within the taste bud. So maybe this is a separate population, or potentially there may be other roles for these multi-tuned signals. So now I want to transition a little bit to talking about my interest in chemical sensitivity throughout the rest of the body. And as Alan had also mentioned, taste receptors are expressed in many different tissues throughout the body, throughout the digestive system, the respiratory system, the cardiovascular system, and even the genitourinary tract. And while not all of these taste receptor expressing cells are signaling to the brain for everything, some of them are. And so this is the type of circuit that I'm really interested in following up. So to this point in my talk, I've really been focused on this fungiform to the geniculate ganglion circuit. And just recently, my colleague Jin Zhang et al. In 2020 published this beautiful paper looking at the sour signaling from the tongue to the brain. But within this paper, she also identified using single-cell RNA sequencing of individual geniculate ganglion neurons potential markers for each of 5 populations of these cells that may be responsible for individual taste modalities within the geniculate ganglion. Just a quick shout out, Jin is on the job market right now, so if you're looking for a really fantastic taste person, she is someone to look into. But as Alan had also mentioned, the fungiform taste buds aren't the only taste buds on the tongue. There's taste buds that line on the surface of the palate, and these both are innervated by the geniculate ganglion. However, there's another very large group of taste receptors right in the back of the tongue within the structure called the circumvallate papilla. And this is innervated by neurons that have cell bodies within the petrosal ganglion, which is part of the petrosal vagal ganglion complex, which includes the nodose ganglion, the petrosal, and the jugular, all kind of smashed together within the mouth. It's a real, Grand Central Station for all of this information funneling from the body to the brain. So my graduate student, Brian Fowler, is very interested in looking at the taste responses that are coming from the circumvallate papilla, looking at the activity of individual petrosal ganglion neurons to taste stimuli. And he's now gotten this prep to work really nicely, and we're starting to collect data, and we'll be able to compare and contrast what kind of responses we see in the petrosal ganglion and how that is similar or different to taste responses in the geniculate from the front of the tongue. Recently, a paper by the Steve Lever's lab was describing these extraoral taste buds within the larynx. So there's actual taste buds that are found within the laryngeal tissues, and his group has shown very nicely that these taste buds are responsible for triggering cough and swallow reflexes when you kind of swallowed something down the wrong pipe, right? That triggers that really aversive cough and swallow reflex to get rid of that blockage of liquid within the trachea to help you clear your airway. And this is mediated by a population of vagal ganglion neurons that are expressing P2RY1. We'll hear much more about these other very, very interesting nutrient sensing circuits within the gut. This was Maya Kelber's report about the identification of neuropod cells with fast synaptic transmission from the intestine to the vagal nerve. And these studies, along with a few others, we'll hear about from Dr. De Araujo's lab and others, have really indicated that this nutrient sensing within the gut is highly important for forming preferences to food and also giving information about the quality and the nutritional value of the contents of the gut. So many other people have identified as well the expression of individual taste receptors within these populations of enteroendocrine or possibly neuropod type cells within the gut epithelium. Some of them contain T1R3, the sweet umami GPCR. Others contain SGLT1, a glucose transporter. Some co-express the both. together. Other cells, including enterochromaffin cells, have been shown to express bitter taste receptors. And while we know that these receptors are expressed in certain subpopulations of cells, we don't really know too much about the connectivity to the vagal ganglion if it occurs. So what I'm really interested in following now is looking at this possibility of vagal ganglion neuron connections to taste receptor expressing cells within the gut. And to help us along with this have been several very, very informative studies looking at these expression profiles of individual ganglion neurons, forming these ideas that there are these discrete clusters of similar cells, but we're still not sure what all of them do. So now it's kind of a race to figure out what is the function of all of these different cell types within the vagal ganglion. Where do they send their peripheral axons to? Where do they send their central axons to? And what circuits are they actually involved in? And what behaviors do they use? So to begin to look at this problem, I think one of the great things that we can do is to utilize some new tools. And one tool that is close to my heart is GRASP, GFP reconstitution across synaptic partners. And GRASP is essentially based off of GFP, the green fluorescent molecule, but we split it into two parts. Let me turn my laser pointer back on. So one half of the GRASP we express on the presynaptic side of a neuron, and the other half of the GRASP we express on the postsynaptic side. If these neurons are not in contact, nothing happens. They don't come close together enough to reconstitute fluorescence. But if these GRASP halves are on the pre and postsynaptic side of a circuit, at the synapse, the GRASP molecules can recombine and form a fluorescent molecule. In my previous work, I've developed many different tools for GRASP, and this has been catching on, especially in the Drosophila field, but also now in mouse. So I made a activity-dependent GRASP system where it's linked, the presynaptic half of GRASP is linked to synaptobrevin, and so only when the neuron is active and vesicles fused to the presynaptic membrane is the GRASP available for synapse, or for reconstitution at the synapse. I also made different versions of GRASP in multiple colors. Starting with the green GRASP, I mutated the GFP molecule into yellow and cyan versions. So now we can look at GRASP in multiple colors. And as I mentioned, many others have been improving GRASP methods for individual model systems, including the mouse, Drosophila, zebrafish, and C. elegans. My PhD student in the lab, Shannon Landon, has been really busy at work creating multiple GRASP lines to investigate chemosensory connectivity of the peripheral sensory system. So we created 3 different versions using back transgenic models, driving the green GRASP using a T2R19 bitter promoter. We're going to make a yellow GRASP with the C1R3 promoter to express it in sweet and umami cells, and a cyan GRASP version using the PKD201 or sour cell type promoter. We also are in the midst of making a TRPM5 YFP GRASP molecule for expressing the YFP pre-GRASP within TRPM5 populations, which in the taste bud include the bitter, sweet, and umami cells. But TRPM5 is also really highly expressed throughout many chemosensory cells in the gut and respiratory system as well. So this might be very, very useful for extraoral chemosensory circuits. In addition, we need to make the post-GRASP. And for us, the easiest and the most useful tool is a pre-dependent adeno-associated virus. So this contains both the post-GRASP component as well as a tomato fluorescent molecule, so that we can really see the small fibers innervating the taste buds and into the peripheral organ systems that we're going to investigate in the future. So unfortunately, maybe I'm going to blame COVID a little bit, but we've got the most data so far on the sour pre-CFP GRASP system, and I'll show you a sneak peek at how beautiful this can look. So Shannon has already characterized our founder line for the sour pre-cyan grass mouse, and we can see that it's a beautifully restricted expression pattern within the taste bud. So this is a circumvallate taste bud with a merged image. You can see how beautifully and tightly packed all the taste receptors are within this bud. We can use an anti-GFP molecule to recognize the presynaptic CFP grass, and it's expressed in a few of these cells, which is very colocalized with the carbonic anhydrase sour cell marker. So we believe that this sour pre-grass mouse really is going to be good at identifying sour synapses between the taste receptor cells and the gustatory neurons. This TRPM5 marks the sweet, bitter, and umami population of cells within that bud. So to do this experiment, we need to have both the presynaptic and postsynaptic sides. So first, we express the AAV synapsin pre-dependent post-graphs construct along with a pre. So we kind of cheat and put the pre in there at the same time. So we can use this to express the post-grafts throughout multiple different ganglion subtypes. And we can do this because the nucleus of the solitary tract is where all of these neurons tend to innervate. And as Alan had mentioned, there's this beautiful kind of rostral caudal aspect to it. So more rostral tends to be more taste, more caudal tends to be more vagal. And we kind of spread it all over the place. So we can hit multiple ganglia, including the geniculate ganglion and the nodus petrosal ganglion here. You can see these beautiful RFP expressing cells, which are expressing the postsynaptic grasp components. And over here, we can see that after a few weeks, we can see these fibers very brightly labeled in the circumvallate papilla, and also we can see it in the foliate papilla as well. This beautiful picture was taken by Zagnita Garcia in my lab, who's now going to be defending her master's thesis next Monday. So if we do this experiment with the in the background of the presynaptic cyan grass sour grass mouse, we can express the postsynaptic grass and see these fibers innervating the bud, which the tomato label here. But most importantly, we can see these beautiful, very bright, but very punctate CFP cyan grass signals, which we believe really represent the bona fide synapses between sour cells and these fibers that are innervating them. In this example, we've used antibody labeling to see the sour cells with the anti-CAR4. But if we don't bother to do the antibody labeling, we can actually see even more of the cyan fluorescent synaptic signals. And we can see within the bud, multiple puncta, which we believe are these synapses between the sour cells and the postsynaptic neurons within the geniculate or the petrosal ganglion. So to just, this is just a promising start of this technique, but I think it has lots of potential. And so our next steps, which we wanted to investigate, is to test the synaptic connectivity and specificity of peripheral taste circuits using our 3 different grass colors, the green for bitter, the blue for sour, and the yellow for sweet, and see if we can identify the postsynaptic partners within the geniculate ganglion, potentially those that have been hypothesized using the molecular profiling techniques. We also want to explore synaptic connections between extraoral taste receptor cells and vagal ganglion neurons. And this would be really amazing if we can identify potentially those neuropod type synapses with cells that express taste receptors in them, and potentially identify different circuits for appetitive versus aversive type signals within the gut. One real advantage of GRASP is that it is fluorescence, and it's fluorescent. It doesn't require any post-processing necessarily. So we can potentially visualize the dynamic process of rewiring of neuroepithelial circuits as cells undergo turnover. One great thing about the tongue is that it's very accessible to imaging, and potentially we could follow these synaptic contacts over time as the cells go through turnover. and then potentially in the future look at how these processes may be modified or disrupted by certain diseases, neurodegenerative disorders, high-fat diet, any of these types of manipulations that can affect taste wiring and rewiring. I'd like to thank my close collaborator, Ho-Jun Lee, who's now a PI at Northwestern University. We continue our collaboration often. My previous advisors, Charles Zucker and Nick Riva, and my entire lab, I really thank them for all of their help in doing these experiments. And I thank also my funding sources, both internally from UTSA and from the NIH and the Brain Research Foundation. And thank you so much for your attention and the opportunity to share my work today. Thank you.
I think we're a little over, but there's definitely time for one question if anyone has one. We can make time. Okay, well, I think maybe we can.
I have one question if we have time.
Yeah.
That was a beautiful talk, and I was curious because you said you're going to look more at how the connections are made in the gut, and obviously that's close to my heart. And I'm wondering because if I remember correctly, SEM3A is expressed in the gut. So are you thinking it might be similar molecules, or do you think it could be different players down there, or it's just too early to tell?
It's too early to tell, but that is something I'm obviously going to look at very closely. If this is a conserved mechanism that helps the peripheral ganglion neurons find their targets, I think that would be quite elegant. But I would be so satisfied if that's the case, but I better look into it. Yeah.
Of course.
Okay, so I think we're gonna break until 1 p.m. And so we're gonna keep the meeting up, so feel free to stay on or log off, whatever you feel, and log back on. But yeah, we'll see everyone. So the way to, if you go on your screen, you can hit this expand here, but I could just do it as the quick time share. I just have to remember to mute myself. and we're going to jump to Diego Boriquez's talk, which is going to be fielded, questions are going to be fielded by Maya Kalebearer. And then at 1:40, we'll get back to Yvonne's talk. Actually, yeah. Okay, so let's figure out.
Okay, so someone's going to be running Diego's pre-recorded presentation from her computer, but I'll go ahead and give the intros. So this is, welcome back everyone. We're now entering our afternoon session of the symposium, and we've reordered a little bit to give Yvonne some time to work out some technical difficulties. So it's my pleasure to introduce Dr. Diego Boriquez, who received his PhD from North Carolina State University. And as a postdoc with Roger Little at Duke University, he was using electron microscopy to identify a really fascinating cell type, the neuropod cell within the gut epithelium. After his postdoc, he did an illustrious fellowship at the Grass Foundation at the Marine Biological Laboratory. And now, as an assistant professor at Duke, Dr. Boriquez studies how the brain perceives what the gut feels, how food in the intestine is sensed by the body, and how sensory signals from nutrients are transformed into electrical signals that can alter behavior. His title today is A Gut Choice. But unfortunately, Dr. Boriquez could not join us live, so he has prepared a recorded talk. And then to participate in the Q&A in his place, Dr. Maya Kalebearer will be fielding questions. Dr. Maya Kalebearer received her PhD in cell and molecular physiology from Yale University, studying sensory neurons innervating the airway with Sven-Erik Hjorth, using RNA sequencing technologies to identify how inflammation affects vagal nodose neurons. As a postdoc in the Boriquez lab, Maya has been using single-cell sequencing and intravital imaging to discern how sensory function in the gut is integrated by the vagus nerve. So we'll now pull up Diego's presentation and run it for us. Okay, I had a lot of trouble hearing that. I'm sorry. I don't know if the audience had similar trouble. But we're going to move ahead, and I'm just going to play the video. There's going to be a small expand view button in the lower right corner that you can use to make it bigger. I don't have control over that for you, so please do that for yourselves in your window. So here we go.
Good afternoon, and thank you for the invitation to this symposium, Lindsay. Unfortunately, I'm not able to be with you guys live today.
Salma, you'll need to mute your microphone, I think.
Professor Dr. Maya Kalebearer will be here at the end of this presentation to answer questions regarding our research. So I brought you here today a book that at some point I read, and it is fascinating how it is that we come to develop scientific knowledge and sometimes overturn some scientific paradigms or expand some scientific ideas. And one of those ways in which we scientists advance fields is by the use of tools. So today I thought that I will highlight how it is that we have been using tools to advance the field of gut-brain sensory signaling. And as Thomas Kuhn put it in his book, the scientist who is striving to solve a problem defined by existing knowledge and technique is not, however, just looking around. He knows what he wants to achieve, and he designs his instruments and direct his thoughts accordingly. And I specifically wanted to highlight this quote because I believe that we are in a period in the field of gastrointestinal sensory transduction in which the development and adaptation of new tools will definitely benefit to understand how it is that sensory signals arising from the lumen of the intestine modulate our behaviors. So we are all here today to talk about this topic of the gut-brain axis. And by definition, an axis is an imaginary line about which a body rotates. And the reason that we call it an axis is because for long we have thought that the gastrointestinal epithelium did not receive direct synaptic inputs from nerves. In fact, the idea is that when we ingest nutrients, there is within the gastrointestinal tract, there are these villi projections that are lined by a single layer of epithelial cells. And when nutrients arrive to that epithelial layer, there is this gut endocrine cell, which senses nutrients and release some neuropeptides that either enter into the bloodstream or affect nearby nerve fibers indirectly through the paracrine action of the hormones. And part of this work or this concept was built upon some evidence, particularly in the 1970s, using electron microscopy at the time. And even though conventional electron microscopy has this unparalleled XY resolution, unfortunately, it is pretty limited in Z, and also the field of view is very narrow. And a year later, after this report in which they were trying a clear synapse, some of the same scientists reported that indeed these cells make synaptic connections. However, the resolution of the image, the lack of context prevented from this kind of knowledge to be expanded upon. As you see here on the upper right corner, we have these large dense core vesicles that are supposed to be these endocrine cells. And then below, the scientists highlighted some synaptic formations with nerves in the lamina propria. So what I thought that I will do is I share with you 5 experiments by which we have used new technology to be able to advance this field of gut-brain sensory transduction by detailing the synapses between the sensory cells, the circuitry, and also the consequences of activating that circuitry to behavior. So the first one is this idea of neuropods, and this was emanated from an observation early on in 2010, in which using a transgenic GFP animal, I was able to observe that some of the sensory epithelial cells had a very peculiar anatomy. And in fact, getting close in here, we have these cells in the epithelial layer, but underneath these same sensory epithelial cells have this very prominent axon-like process that eventually I gave the name of a neuropod. And when we began studying this field, we began using the tools that we were familiar with. And one of those was electron microscopy. And thanks to the emergence at that time of serial block phase scanning electron microscopy, or 3D-EM, we were able to reconstruct the entire microanatomy of these cells. And essentially, the way that 3D-EM works is a scanning electron microscope that is outfitted with a diamond knife. And in this way, the tissue is placed in this stage, and it can be shaved off very thin slices while the microscope proceeds to image that surface. And by doing that multiple times, one can reconstruct at the ultrastructural level the microanatomy of the tissue, and by that, the biology of these cells. So here is a reconstruction from a neuropod cell. There are about 510 slices that have been reconstructed in there manually. And we have in green the neuropod cell that is separated from the basal lamina by these subepithelial myofibroblasts. And in cyan, we have a cell of the enteric nervous system. In this case, is enteric, and this is an enteric glia cell that is extending a arm to reach out the neuropod. And this was the spark of a series of experiments that for us really expanded our imagination, because it showed us that this cell not only has this very unique feature, but is also through that feature interacting with other cells, specifically those cells of the enteric nervous system. So the second thing that we observe is that these cells indeed form a synapse. So now that we have the ability to identify those cells by fluorescence, we were able to use immunohistochemistry. And as you can see here, some of those neuropod cells clearly are in contact with some nerve fibers, as observed here. This is a slice of the colon as these transgenic GFP reporter mice labeled primarily those neuropod cells of the colon. And this is a PYY GFP transgenic mouse. Here is another example in which we have this green cell that is extending a small basal process, which is contacting this neurofilament medium nerve fiber, and that has been double stained with calcitonin gene-related peptide in cyan. So in red is this neurofilament medium fiber, and in cyan is this calcitonin gene-related peptide staining. So at some point, I remember presenting some of these results, and a scientist brought up to me that they have also observed something similar. And these are some cells that have been fixed from the rat intestinal epithelial layer and stained against serotonin. So these are the so-called enterochromaffin cells. And as you can see here, these cells also have beautiful processes, these neuropods. Here's another example. These have very elaborate processes. And here we can see the cell is still attached to a nerve fiber, even though it has already been enzymatically digested and isolated, showing that indeed these cells are contacting nerve fiber. And in some estimates using immunohistochemistry, we have found that anywhere from half to 2/3 of these neuroepithelial cells seem to be in some sort of contact with nerve fibers. But the question is, do they actually have the ability to connect with a nerve fiber? Because the contact doesn't mean necessarily connection. And this is a very important distinction, because the connection is the ability to not only form a synapse, but also transduce a sensory input, convert a chemical input or mechanical input into an electrical pulse that can be recorded from a neuron. So in this experiment, we have on the left side a neuropod cell that has been isolated using a CCK GFP transgenic mouse. And on the right side, we have a sensory neuron in red, as observing here, that has been enzymatically digested and isolated. And when they are put in culture, these two cells from the gut and the brain, look what happens. So not only they are able to recognize each other, but they are able to reconnect in vitro after they have been isolated from the body. And this was a really eye-opener experiment for us, because it was the beginning of a series of new findings in which we were able to document not only the circuitry, but also how it is that these cells begin to communicate. Of course, there are like several things that we don't understand in there. Like there are multiple open questions, like for instance, the fact that the sensory neuron is able to regenerate within a few hours and reconnect with those neuroepithelial cells. And it seems that at least the video suggests that the neuroepithelial cells are guiding the regeneration. But those are still open questions that are waiting for new investigations. So because we had this platform, one way in which we tested transduction was, can we isolate the sensory neurons, and what will happen if we put glucose stimulus into the bath? And what we found is that these vagal nodose neurons do not respond directly to nutrients. For long, it was thought that the vagus nerve itself is a nutrient sensing nerve. However, this data is suggesting that the vagus nerve works more like an interneuron, in which it relies information from one side to the other and also integrates information to be able to provide feedback. However, when we culture this nerve fiber, this vagal nodose neuron with a neuropod cell, not only they make synaptic connections, but now when they are stimulated with a glucose stimulus, the vagal nodose neuron responds electrically to that glucose stimulus. And this is important because it is the basis of sensory transduction of a sense, the ability to convert an input, in this case a chemical input from glucose, into an electrical pulse that through a synaptic connection has been relayed onto the vagal neuron, from which we are recording these excitatory postsynaptic potentials and the action potentials. So now that we had the basis, the basis of sensory transduction, the next question that we had was, what is the circuitry that these cells are forming? Because depending on the circuitry that they are forming, depending on the neurons that they are connecting to, then perhaps we could begin understanding what it is, the type of behaviors that these cells are guiding. So here I took inspiration from this beautiful paper published in 1979 by Professor Francis Crick at the time at the Salk Institute, in which in this Scientific American article he titled Thinking About the Brain, he wrote about new ways in which we will be able to understand better the brain if we develop technologies. And two technologies that he highlights in this article, or ideas about technologies, is one is a technology that will enable us to untangle synaptic connections between neurons. And perhaps he ponders in there about ways in which we could identify how it is that two distant neurons could be connected by one synapse. And in 2007, it was published the first paper showing that the neurotropic virus, rabies virus, could be used to untangle these synaptic connections between two neurons. So in a leap of faith experiment, we use this monosynaptic rabies virus in which this virus has been genetically modified, so it does not have the ability to jump from synapse to synapse, but it has the ability to label the first entry cell. And when we put a bolus of this rabies virus into the lumen of the colon by enema, 7 days later, this virus infects a specific type of epithelial cell, as you can observe here. Now, I should highlight that this experiment has a few details that are important to take into consideration, because in the future, perhaps they could be addressed by other investigators. One is that the mice need to be very young. So these are P1 to P3 mice. And the reason is that, perhaps one of the reasons that we think that the virus only works in very young mice is because as the mouse, as the gastrointestinal immune system develops, then there are some cells that could clear that virus. And other scientists have suggested the same thing in peripheral neurons, particularly on somatosensory neurons. So the mice used for these tracings are very young mice. The other thing is that the titer of the virus needs to be pretty high. So this is the least that can be used in here is 10 to the 9 particles. And also the volume is much higher than what one will expect to use in the brain. So while in the brain, one will use 300 nanoliters per mouse, in this case is anywhere from 5 to 10 microliters per mouse. So we're talking about 20 times more the volume, at least, per mouse. However, the importance of this experiment is that it showed that the rabies virus could be used to track the circuitry. And in fact, by engineering the complementary RABG gene inside of these neuropod cells, now we have the ability to place this delta G rabies virus in the lumen of the colon, and the virus will infect this neuropod cell, recombine with this RABG glycoprotein, and spread through synapses onto the nerves. And if this is the case, then we should be able to observe a direct synaptic connection with a nerve fiber. And indeed, this is what we observe. Here is a cell that has been stained by the PYY antibody in red, and it still has remnants of GFP from the rabies virus that has spread onto this nerve fiber, as you can see in here. And what I usually like to say in here is that literally this is the point where the gut meets the brain, because here we're talking about the epithelial layer from the gut, and here there are some nerve fibers that are right underneath the intestinal epithelial layer, and some of those nerve fibers go up into the vagus nerve, as observed here. So these are 3 vagal nodose neurons that are monosynaptically connected to those neuropod cells in the colon. This is a reconstruction in 2-photon microscopy. And the point is that there are 2 points that I would like to make in here, that by tracking this circuitry, we found 2 components of the sensory circuitry. One was that anatomically, the basis was there for the gut to be able to transduce a sense, just like the nose, just like the tongue, just like the eyes, just like the ear. So the gut has the anatomical basis for sensory transduction. But the second thing, inadvertently, by the use of this rabies virus, we stumble upon the possibility that if a neurotropic virus would access one of these neuropod cells from the lumen of the intestine or the lumen of the colon, the virus could jump one synapse and spread to the brain via these synaptic connections. And this, I believe, it is important because, as you may have heard, there is quite a bit of interest in neurodegenerative diseases and how it is that they spread from gut to brain. However, the circuitry and the paths are still unclear, and certainly this gut-brain connection provides a tantalizing path for pathogens to spread from gut to brain. And here in this other experiment, this is actually a tracing from the proximal small intestine. So now the rabies has been put by gavage into the stomach of the, intragastrically into the stomach of the pup. And some of that rabies has accessed the small intestine, in which it has infected these CCK, CRE, RABG, TBA neuropod cells, and then spread via the vagus nerve to the nucleus tractus solitarius. So here you have some nerve fibers that are projecting into the base of the brainstem. And in fact, this nucleus tractus solitarius is one of the areas that controls the pleasure of nutrients, which Dr. Ivan de Araujo has expanded upon the subject in a previous presentation. So we have a cell that responds to a nutrient that forms a synapse, that connects to a cranial nerve that potentially sending those signals up to the brain. So the question is, what is the transmitter that is being sent? What is the transmitter that is being used to send those signals up to the brain? So we use, to answer this question, we use rather classic technology, vagal calf recordings, in which 2 electrodes are placed in the cervical vagus right below the nodos ganglia. So those 2 electrodes are placed around the neck of the animal, and the nutrients are perfused into the lumen of the intestine while vagal nerve activity is recorded. So if sugar is perfused into the lumen of the intestine, as you can see here, there is a rapid rise in vagal nerve activity within a few seconds. Vagal nerve activity increments. However, by using optogenetics, we were able to determine that indeed these neuropod cells are necessary for the sensory transduction of sugars from the lumen of the intestine onto the vagus nerve. So when we silence using halorhodopsin these neuropod cells from the proximal small intestine, the CCK expressing cells, then the vagus nerve does not respond to the glucose stimulus. And in fact, in subsequent experiments, we confirmed that the cells that express the electrogenic glucose transporters are these neuropod cells and not the vagal nodose neurons. So the signal emanates from the epithelial layer. And quickly, the next question was, would these one of these neuropeptides will be the signal that is transmitting this electrical excitation, like cholecystokinin? However, when we block cholecystokinin receptor A in the periphery using this the vasepite blocker, we found that the first couple of minutes of that vagal response still remains intact, although after 2 minutes, most of the vagal response is ablated by ablating the CCK signaling, indicating that CCK is responsible for the delay onset of this stimulus. However, the first 2 minutes where there was another transmitter that could be responsible for the signal. So in using, fortunately, microscopy has evolved tremendously in the last 10 years, particularly electron microscopy. And here we have a type of electron microscopy that was developed by my friend David Hildebrand, now at Rockefeller University, and also Professor Wei-Chun Lee at Harvard University, in which they outfitted a transmission electron microscope to be able to collect thousands of images and collate them together. So here we have a collage of 35,000 images of the colon at a very high resolution. And when we go very deep into the tissue, we're able to observe that indeed these cells have, these neuropod cells have these large dense corpuscles, but they also have these small, clear synaptic vesicles, as you can observe here. And we quickly were able to identify that one of the neurotransmitters used by these cells is glutamate. As you see here, not only they express vesicular glutamate transporters, but they also have the proteins for that. And indeed, using a glutamate sniffer expressed in hex cells and adding a co-culture with neuropod cells, we were able to determine that indeed these neuropod cells release glutamate in response to glucose. So when we add glucose, that glue sniffer is activated, as you can see by the change in fluorescence. So finally, we have this circuitry So we have a sensory epithelial cell in the periphery exposed to nutrients in the lumen of the gastrointestinal tract. It is forming a synapse. It releases glutamate in response to sugar. It connects and activates these vagal nerve fibers that project into the brainstem. What is the behavior? That's the crux of the matter in this case. So for this, we knew for years it was known that there was a post-ingestive effect of nutrients entering the intestine. They had this reinforcing effect when they enter the intestine. And in fact, Professor Ivan de Araujo found that even in the absence of taste receptor, animals are still able to differentiate sucrose from water, but not sucralose, just carrying sweet taste from water, showing that there is an additional signal for sugar to be recognized by the body after ingestion. So we outfitted a commercial phenotyping machine. This is an automated phenotyping behavioral cage in which we can track a food intake, food choice, body weight, and locomotor activity. And when we give an animal the choice between sugar and sweeteners, sucrose versus sucralose, a naive animal takes about 24 hours, but within 24 hours, the animal learns to recognize what is sugar versus sweetener. But thereafter, the animal, even if you change the animal from cage to cage and you show the animal the different solutions, the animal learns to discern a sugar from sweetener within a couple of minutes. And this perhaps is evolutionary an important behavioral response of the animal to be able to discern that sugar that carries nutritive value. So the second thing was to use optogenetics to be able to control the sensory function of these cells. And as you know, optogenetics has been a major advance to be able to control cells within a neural circuit. And then here we have some cells in the lateral hypothalamus that are being stimulated so the animal will eat, and when they are turned off, the animal stops eating. So we work to outfit and to bring optogenetics into the gastrointestinal tract. And this was a multi-year effort that eventually one of the biggest hurdles was that the fiber optic cables that are used for delivering light into the lumen of the intestine are stiff silica fibers that can puncture the small intestine. But but working with Professor Anikieva, Polina Anikieva at MIT, we were able to determine, and she had developed these flexible fiber optic cables, and these fiber optic cables do not puncture the intestine. And as you can see here, they are fully compatible with the delicate intestinal lumen. So we outfit them for through surgery, and these fibers deliver light specifically in the proximal small intestine. And after surgery, this is in an animal 5 days after surgery, the animal has recovered and it is fully functional, just like the others. So now we are able to run the experiment, and in a control experiment with laser that does not activate and this channel, this halorhodopsin, the animal is still able to recognize sugar from sweetener. However, when we provide the correct light and we silence those neuropod cells, the animal is incapable of recognizing sugar from sweetener, essentially rendering the animal blind to the caloric sugar. And this same behavior is replicated by exposing, by blocking glutamate signaling from the lumen of the small intestine. We have found in control experiments that this is not due by a change in consumption. Total consumption after an hour, it remains the same. What it changes is the actual ratio of the solution. So the animal seems to be incapable of discerning the two solutions. I believe that this opens a new opportunity to understand how it is that the subliminal sensations arising from nutrients and perhaps other stimuli within the gastrointestinal tract modulates our behaviors even below the threshold of consciousness. So with that, I would like to thank my team, specifically Maya Kelber, who was the leader of the first work on the circuitry for nutrient sensory transduction, Margarita Klein, who has worked extensively on the rabies viruses platform, Kelly Buchanan, who outfitted the behavior and did most of the sugar preference work, and also Laura Ruprecht, who has worked extensively on the behavioral paradigms. So with that, I would like to thank you very much, and please support us and follow us at GutBrain.
Great, thanks. So I think Dr. Maya Kelber will be able to answer a couple of questions if we have any from the Q&A. It usually takes a while for people to type in their questions, but I found one of the things that was really fascinating was that video of the enteroendocrine cell and the vagal sensory neuron in culture, and then just reaching out and touching each other. That's really the coolest thing.
Yeah, it really is. And there's another video that we have as well where you see the neuropod cell kind of grab onto an axon, and then it just goes along as it keeps growing out. So they certainly have an affinity for each other in the culture system. Yeah.
While we're waiting for anyone to plug in their questions, does any of the panelists have a question for Maya?
Yes, that was really interesting, and I was particularly interested in at the end where with the unpublished data where you're doing the photoactivation in the gut and preventing the sucrose. That was really cool. And I noticed on the next slide, and I apologize if I missed something, you Diego also then showed the effect of blocking the glutamate, but I don't know if one thing that wasn't clear to me, it looked like both sucralose and sucrose increased under those conditions.
Yeah, so, right, and I don't know for everybody else, but the sound was off a little bit, so I think that the slide showing up in Diego's voice were off a bit. But yeah, the very last experiment, we did intraduodenal perfusion of a low dose of these glutamate blockers. So this is a concentration that if we were to give it IP has no effect. And it's not that both increase, it's actually that sucrose comes down and sucralose goes up. So their total consumption stays the same. They're just now drinking equally from the two solutions.
Okay, thanks. Thanks for clearing, because I found that very interesting.
Yeah.
Maya, that was a wonderful talk by Diego, and it's really fascinating data. Did I understand correctly? He said that discrimination occurs by about 3 minutes after they start drinking.
Yeah, so it happens within the first couple of minutes, and so the discrimination seems to happen if you watch the videos. Like we're showing average traces. If you watch the videos really closely, the mice are always given the 2-bottle choice, and they usually they drink one, they go and they drink a little bit of the other, and then they groom themselves a little bit, and then they go on to like decide what it is.
Because I'm wondering about the dynamics of gastric emptying, you know, because 3 minutes seems, you know, really quickly. Now, I think, you know, I'm not an expert on gastric emptying, but I do believe that there is like sort of this initial dump, and then it starts getting regulated depending on the concentration of the sucrose. So I'm just curious about the temporal dynamics of all that in terms of the control of the behavior, and that's why I asked.
Yeah, and right, we've also had that thought as well, and we think that there's like, yeah, that initial leak gets a little bit of what each solution is before it makes it. And then there's probably goes on to do all the gastric emptying that's based on caloric content and things like that.
Because also, and the other side of it is that, and not to dominate the time here, but that, you know, because the animal has to somehow be able to discriminate which solution is leading to the positive signal, right? And so a lot of that has to go on. And so it's truly amazing that the system that the animals can actually make that discrimination within such a short period of time.
Yeah, and there's been a lot of work by Sclafani. So we do our training with 2 bottles. They are always exposed to the 2 bottles at the same time. And Sclafani usually trains 1 bottle at a time. Mm-hmm. So it's possible that when you only have 1 bottle, they're also making maybe some oral discrimination as well. But at least in our hands, in our assay, we find that it's really just, it seems to be just at the gut where they finally are able to distinguish the 2.
Okay, well, I think we're running a little short on time, so we'll wrap up the Q&A for now and move on to our next speaker, Ivan de Araujo, who I think has got his technical glitches all fixed up, so we should be prepared for his talk. Again, like if there's questions that come up along the course of the talks and we don't address them directly in the Q&A, please keep them in mind for the panel discussion at the end of the day. All right, thanks, Maya. So now to introduce Dr. Ivan de Araujo. Dr. Araujo majored in philosophy and math at the University of Brasilia. He completed a doctorate on functional neuroimaging of taste representations, supervised by Edmund Rawls at Oxford University. And for his postdoc, he worked with Sidney Simon and Miguel Nicolelis at Duke University, where he used electrophysiology to characterize the neural populations mediating appetite and satiation in rodents. From 2007 to 2018, he directed the neurobiology of feeding lab at the John Pierce Laboratories at Yale University. And in 2008, Ivan was the recipient of the Young Investigator in Gustation Award presented at ACHEMS. More recently, he moved to Mount Sinai School of Medicine, where his laboratory is currently studying the modulation of brain dopamine systems by gut hormones and the mechanisms used by brain cells to sense nutrients. I'm very much looking forward to his talk today, entitled The Vagus Nerve and the Physiology of Reward.
Thank you. Let me just make sure. You can see my screen, right?
Yes, we see it, but we see the whole thing, not just the presentation.
Yeah. So let me. I'm trying to go back to a PowerPoint. Okay. Is that okay?
That looks great.
Okay. So thank you so much for the invitation, for allowing me to participate in this great symposium, and I'm looking forward to the discussions in the end. So as a matter of introduction that I should have done just before Diego's talk, I would like to start by mentioning that over the last 100 years or so, we developed a quite robust or rather well-accepted model for how food intake is controlled, right? So over the years, we formed this conception that the hedonic value, that the rewarding properties of food are derived from the sensations that arise from the oral cavity, taste, aroma, texture, and so on. And these inputs from the oral cavity and the nose, they travel to the brain via cranial nerves and stimulate our reward systems in a way that is that lacks in and by itself some sort of regulatory or negative feedback. And on the other hand, we seem to be endowed with a negative feedback system that arises mainly from the gastrointestinal tract. So the idea is that the gut can put brakes on this taste-driven reward system by providing some negative feedback to the brain. So sensations like fullness or bloating or even nausea would work to control the amount of overeating we may be subjected to when presented to rewarding foods. So in this system, the vagus nerve, which is the longest and probably best studied cranial nerves in vertebrates, would play a crucial role. You would, via its innervation of the gastrointestinal tract, it would provide this negative feedback to the brain and control and downregulate food intake. So we have this conception well formed in our scientific culture and even in the popular culture. But however, if you look carefully at the data that has been generated over so many years, the results do not show to any extent that damaging the vagus nerve, this long nerve innervating the gut and other visceral organs, would cause overeating. So vagotomies and other manipulations rarely in and by themselves induce overeating in animals. And to make things even more complex and interesting, it has been found empirically over the years that the stimulation of the vagus nerve happens to be a very strong modulator of emotional and affective states. So, for example, the stimulation of the vagus nerve at the trunk level, at the neck, at this level. For example, it's one of the few FDA-approved treatments for medication-resistant depression. There are some patients who are in a more serious condition, suffering from suicidal thoughts, for example. They respond positively to this treatment, showing that the nerve, more than providing some sort of negative feedback from the body, seems to be involved in some more complex, affective, decision-making related functions. So the question really is why the data over the years about the function of this nerve is so complex and in many cases contradictory. And I believe this is basically essentially a technical problem related to the complexity of the nerve, related to the anatomical arrangement of the nerve. So what we call the vagus nerve, here represented in the rodent, which is the best studied model so far, the vagus nerve, these long fibers connecting the brain to the body, is actually related to two different populations of cell bodies in the organism. On one hand, we have sensory neurons located in the periphery forming what we call the nodose ganglia, which is a typical peripheral sensory ganglion that is located outside the brain, and they are pseudopolar, so they send one axon towards the organs and another one terminating in the nucleus of the solitary tract that we heard so much about today, and Alan gave a very nice introduction to the nucleus of the solitary tract. So we have these sensory fibers here connecting the organs to the brain. But on the other hand, we have a second population of cells that we call the parasympathetic or the motor vagal neurons in the dorsal motor nucleus of the vagus that are located in the lower medulla just next to the nucleus of the solitary tract. And these cells are parasympathetic, and they send their axons towards the body to modify their function, to modify organ function. And the tricky thing is that these fibers leaving the brain, they cross through the nodose. They don't connect to the nodose neurons, but they cross through the nodose on their way to the target organs in the periphery. So given this arrangement of these two sets of fibers running together towards the body, you can appreciate that cutting the nerve or stimulating the nerve will inevitably affect both sets of neurons simultaneously, in addition to affect different organs at the same time. So this is a technical challenge that had to be overcome in order for us to really understand what the nerve does. So what we have been trying over the years is to use more modern neurobiological tools like optogenetics and viral transfections and so on that you've been introduced to during Diego's talk, to try to get a more specific handle over the function of the nerve. So I'm going to explain here using a cartoon roughly what we have, how we have approached this function, specifically how we approached our research question about the function of the sensory vagal neurons innervating the upper gut. So here you have a representation of the mouse. So when the mouse eats something, this is going to be the presence of the nutrients will be sensed by these vagal terminals here shown in yellow, right? So the cell bodies are here in the nodose ganglion. The nodose ganglion has a number of neurons running through, as I mentioned, towards the body and synapsing here in the nucleus of the solitary tract. So the way we targeted specifically the nodose neurons innervating the upper gut was by combining using a combinatorial viral approach in the following way. So we first injected a retrograde virus carrying Cre recombinase in our target organ, in the upper gut in this case. And Cre traveled back to not only the nodose neurons that innervate this organ, but also to other neurons innervating the upper gut, in particular the parasympathetic neurons. So to specifically target the sensory neurons, we administered a second injection, in this case, a Cre-dependent channel rhodopsin carrying virus, right? So by targeting the upper gut with the retro-Cre virus, and in addition targeting the sensory ganglia with a Cre-dependent virus, we exclusively transfected the sensory neurons that innervate the upper gut, leaving other organs unaffected, and importantly, leaving the parasympathetic neurons unaffected. And to complete the protocol, we implanted a optical fiber over the nucleus of the solitary tract to take advantage of the fact that the terminals from the nodose will also express channel rhodopsin. So using this preparation, we can perform in vivo experiments in awake animals by optically manipulating gut innervating neurons exclusively. So because this was a kind of a new preparation, we didn't know really what to expect. So in the first experiments, we performed some simple protocols. In this case, you have here a behavioral box where animals have the choice between poking between two holes, right? So in one hole, if they poke here, nothing happens. And in this case, if they poke here, the light will be delivered to the terminals innervating the gut. So if the animals are unhappy with this stimulation, you would expect they would never come back to it, right? But interestingly enough, what we observed is that the animals actually not only self-stimulated a few times, but kept coming back to the hole that was spared to light stimulation. So they were self-stimulating via the activation of the gut innervating neurons, right? So this is a typical session. The animal hangs around, try the two holes, but over time they very robustly accumulate poking into the hole associated with light delivery, right? So behaviorally, this type of behavior is interpreted as meaning that the animals are motivated to obtain the stimulus. And in fact, this is very similar to the classical protocols discovered in the 50s by James Olds and his colleagues, where he initially accidentally found that animals will self-stimulate certain populations of neurons even in the absence of any physiological deprivation like hunger or thirst. So animals, if you put the electrode in the right place, which he mistakenly did in the beginning, and had the insight to notice that the animals were attracted by the stimulation. So animals will even give up food, sex, and water in favor of stimulating these certain populations of neurons in the brain that turn out to be dependent on dopamine transmission. But anyway, this was a revolution at the time, because then it meant that physiological deprivation was not required for reward. Just certain brain neurons have the power to drive the behavior of the animal in a somewhat autonomous way towards reward. And here, if we look at our data, you can see that over days, animals increase the number of pokes on the hole that delivers the stimulus to the gut innervating neurons. This happens, they will choose the hole associated with light even under extinction, meaning that the laser source is off. And this is a very typical self-stimulating behavior, very similar to what you see when animals self-stimulate the brain. Also, animals will develop preferences for areas of the home cage when you associate them to vagal stimulation. For example, animals normally like to spend their time on the bedding area rather than, say, sitting on a flat surface like this. So you can see the animals will spend most of its time on the bedding area that is preferred over this flat surface, right? So this is a fast movie. The animal is not crazy, but in a moment we are going to pair the presence of the animal on this flat surface with laser stimulation, and you are going to see that the animals will change their preference towards a less preferred area whenever this presence on this less preferred area is paired with vagal stimulation. So you can see now the animals they hang around, they go to the bedding area, and they decide to go back, and they clearly would rather stay in the flat area rather than the bedding, because now we are pairing the flat area to vagal stimulation. And another example is that, so this is the data for place preference is clear. And also, when we pair lichens to a non-caloric flavor with vagal stimulation, animals will develop a preference for this non-caloric flavor over another one that was equally preferred before, whenever we pair the lichen of this flavor to the laser, right? So we have a device where the lichen triggers the laser. So in this case, this pairing is sufficient to produce not an extreme, but a clear preference for certain flavors that have been paired to the laser. So finally, we also noticed that vagal stimulation is going to cause the release of dopamine, which is the fundamental transmitter for reward processing in the vertebrate brain, on areas associated with reinforcement learning. So here is a side view of the rat brain, right? The eye would be here, here's the back. So these populations of dopamine neurons that release via their axons dopamine in the striatum, which is an event absolutely fundamental for reinforcement learning and decision making, we found that stimulating the gut innervating nodose neurons will cause a significant increase in the levels of dopamine in the striatum in a way that is very similar to what we observe if we put caloric food directly in the gut. And interestingly enough, this was very robust for the right nodose, and this was the same for the behavior rather than the left nodose, which was something that we didn't expect. We need to explore further, but there is some, let us say, asymmetry in this system. So to summarize, we believe that these nodose neurons that innervate the vagal, innervate the upper gut, they are, or they comply with the formal requirements to fit the description of what is classically known as a reward neuron, because they sustain self-stimulation, as I mentioned. They condition stimulus preferences like flavors and place, and they induce the secretion of dopamine in the striatum. These are the classical, let us say, requirements or conditions for certain neurons to be named or considered reward neurons. So over decades, starting with James Olds and colleagues, they mapped the location in the vertebrate brain of the reward neurons, starting with the rat. Then they found this the analogous populations in larger animals like cats and non-human primates. And using imaging and stimulation techniques, we know that essentially the same areas exist in the human brain. And we propose that these reward neurons, they are not confined to the central nervous system, but they actually exist outside the brain. In particular, peripheral neurons that are nutrient sensors seem to be a fundamental part of this reward system. And in fact, if you think about it, nutrients, food is the ultimate reward animals must procure in order to perform any other activity. So we believe that the brain reward system is actually anchored on peripheral sensors that actually are the most fundamental type of reward neuron. So just to conclude, we aimed at describing the pathway taken from the gut to the reward system somewhat step by step. And we had to do this because the nucleus of the solitary tract, which is the recipient of these gut terminals, does not project directly to the reward system. So we attempted to know, to describe how the gut information travels to the brain. And I'm going to do a 1 minute summary, so it's a long story. We can discuss the details if you like, but essentially by using cre-dependent strains of anterograde and viruses that travel onwards polysynaptically and confirming these results using cre-dependent retrograde viruses, we delineated a pathway that we believe starts in the gut, in the upper gut, so the vagal terminals, especially the ones originating from the right nodose ganglia, then these neurons as expected, synapse in the medial part of the nucleus of the solitary tract. From there, they innervate the dorsolateral part of the parabrachium in an area that is separated from the sickness neurons in the external lateral. So these are the neurons that Richard Palmiter characterized as related to sickness, and they do not seem to be associated with vagal inputs, at least from the gut. And we found that these neurons here, these glutamatergic neurons, innervate the dopamine areas rather strongly. They provide very strong glutamatergic input to the dopamine system, which then releases dopamine in the dorsal striatum, where reinforcement learning, decision-making, habits, preferences are formed in connection to the cortex. So we have a somewhat straightforward glutamatergic pathway is starting with the glutamatergic nodose neurons to the NTS into the parabrachium and into the classical reward of a system of the brain connecting the gut to these areas. Right. So I would like to thank my my current collaborators, especially Wenfei, and who has been a driving force behind this, with Luis, who is now in Mexico City, and Matthew, and all the other members of the lab, and my past collaborators who I had the pleasure to work with over the last couple of years. And I'm happy to answer any questions.
Thanks, Yvonne. Very exciting talk. We have a nice group of dopamine signaling groups here at UTSA, and so I was so excited when I first read your paper that maybe I could join the dopamine group, because now I'm interested in this gut signaling circuit. So yeah, thanks to you. Now I can be an honorary member of the dopamine club.
Yeah, they are all, I think they are the same pathway in the end, but anyway, yeah.
Yeah. Do any of the panelists have a question while I wait for some questions from the audience?
I have one. That was terrific talk, Yvonne, and I mean, it's really just very elegant work. So, you know, you very convincingly showed that the optogenetic stimulation of those vagal neurons can, you know, drive and reinforce behavior. And I'm just wondering, and I may have missed this, but so, you know, where is the population, or is there not a population of vagal neurons that go in the opposite direction, you know, that would be responsible for, say, satiation? And you know, so I'm trying to figure out how you know, distinguish between those two opposite pole type of neurons.
Yeah, this is a great question. So part of the answer is that we could have been more specific in our experiment. So we know that these neurons drive reward and they produce satiety, even though when we ablate them, we don't see any overeating. The animals become incapable of learning flavor preferences. I didn't have the time to show the data, but that's what happens. But they don't overeat. They maintain their body weight. However, when you stimulate the neurons, they tend to eat less as a sort of satisfaction, right? One possibility is that because we injected both the stomach and the duodenum simultaneously, it may be that the satiety effect derives from stomach inputs, whereas the nutrient sensing that goes on in the duodenum and is sensed by vagal neurons innervating the duodenum provide this reinforcement signal that the animal uses to learn new conditions. And so one next experiment would be to do this whole thing again by targeting the stomach and the duodenum separately. And I put them together initially because I was not so optimistic about the number of neurons we could infect. So I decided to do a big injection. And in retrospect, it may have not been a good idea But that's one possibility. The other is that what we understand by satiety, the other possibility is that what we understand by satiety is reward itself, is like a state where the animals no longer feel the need to procure the reward because the system has been refilled, or so there may be some more behavioral concepts that we need to develop linking satiety to reward in a more intrinsic way. But as a first guess to this question that is very good, I would say that maybe if we separately affect the stomach and the duodenum, we may see divergent behaviors, satiety more or less independently of reward and so on, which is what happened when we stimulate the left nodules. The animals decrease eating, not dramatically, but they do. But they don't seem able to develop preferences or to release dopamine. And we don't know exactly why. We didn't expect that, but that was the case, yeah.
One follow-up question I have is the amount of stimulus you provide to the mouse, and maybe this goes along kind of with how many neurons you have expressing the channel rhodopsin, but yeah, do you need to titrate your stimulus a little bit so as not to overly induce these neurons, or do you find any differences with the stimulus frequency or amplitude?
No, there is a range that worked, and so these channels, right? They don't work in a exactly linear manner, right? So they are artificial in this sense. We didn't need to blast the neurons with activity. There is a range of frequency that worked, and it worked well with dreads. Actually, that's how we started. We transfected the neurons with dreads, and we did a place preference learning paradigm, and that's how we first saw the preference effect. So it seems that if you depolarize them in a reasonable way, they as long as they release some glutamate in the NTS, I believe that the signal. So we see, say for example, when we do these things, we look at FOS in the NTS, right? And there is like a kind of pretty standard pattern of activation, kind of comparable to what we see when we put, like, say, fat in the stomach or something like that.
Yvonne, I have a question. Is the left-right asymmetry borne out in any way by the topology of the actual circuits, or do they innervate differently on the left and right, and are the sensory motor ratios different?
Yes. So yeah, it's important to keep in mind that the visceral organs are asymmetric, right? So we have one stomach turning to one side, and the liver is here, the spleen is there, and so. And in fact, we know from classical anatomy that these terminals, they don't innervate the same regions. Like, for example, the portal vein and any region near the liver, it's almost exclusively innervated by the left nodules, whereas like some parts of the intestine are more specifically targeted by the right. We and others have been noticing that the lower gut seems to be almost exclusively innervated by the right nodules, which has some interesting implications for microbiome action on the brain. So that in the periphery, they are not symmetrical. That is already known. So it kind of makes sense, if you think about it, that they will affect different parts of the brain, because different organs are telling different things to the brain, right? So that's yeah. So that's how we interpret this asymmetry. It's like it's like different visceral organs trying to tell different things to the brain, and then they use two different pools of neurons that go to slightly different areas.
Selma, we have a question from Anand Kulkarni. Could you allow unmuting for them to ask the question?
Am I supposed to do anything? No.
No, no. We're waiting to unmute, but I don't know if it's going to work or not. Sorry. Selma, you're also muted, so I can't hear what you're saying.
I'm having some difficulty with the unmuting part, so maybe we should just read it.
I'll just ask the question directly. So this is from Anand. Have you tried helorhodopsin in the no-dose? Do you expect aversion, or would there be no effect?
Yeah, that's a good question. Actually, I should have mentioned this to Alan too. So we did the inhibition, and we also did something more radical. We used this retro-Cre combinatory approach to eliminate the gut innervating neurons. So we didn't see any effects on aversion. So we did a flavor aversion learning by injecting lithium chloride in the gut. And the ablated animals, they learned to avoid the lithium chloride paired flavor in the same way. The FOS patterns in the brain were very similar. And if anything, the animals lacking the vagus looked more sick than the controls. So as far as gut innervating neurons are concerned, we don't see any obvious relationship between these gut innervating neurons and the ability of the animal to develop aversion. So we see clear deficits in reward learning. For example, when the animals have to do an operant task to get intragastric infusions to so the action is conditioned by the food in the gut. When we do this, when we eliminate these neurons, the animals cannot learn that the food they are getting in the gut is rewarding. However, we don't see any obvious effect on aversion.
What, if I may, can I jump in just a second? One issue about the lithium chloride, it was a good idea and very clever, but of course, once the lithium chloride gets absorbed, it can get to the AP and, stimulate it that way. So it might be interesting to find some other aversive agent that would be acting, primarily through the gut and redo that experiment.
Yeah, that's a good question. There is a compound, the name is escaping me now, but it's supposed to be very aversive and acting on the gut, but the animals looked pretty horrible when we injected it, so we gave up. But it's still, I think that I totally agree, there is a systemic effect, but there was no loss of, there is no partial loss of aversion. Right. So, and I think to be frank, there were papers in the past looking at the effect of lithium chloride on vagotomized animals, and they came to the conclusion that the area postrema is the big player there.
Great. Well, I think this wraps up the questions for now. We're going to have a brief break for 10 minutes, and we'll resume again at 2:30 Central Time, where we'll start off our next panel talks with Suzanne Appleyard. So please go ahead and take a break, and we'll resume in a little bit. Thanks.
Okay, super.
How do is my screen still up?
Yes, it is.
Yes, it is.
So I'm trying to go back and.
You can hit stop share, and then it will.
Yeah, I'm trying to actually go back to the web. Now I think it's fine.
Okay, great.
everybody back? I don't see Selma, but
I'm
she's
here.
still there.
Sorry.
Oh, okay.
Hold on.
Okay.
Is the slide presenting as it
Yes.
should? I was having, for some reason, my computer decided to have a little fit, and I'm not.
Breakdown.
Sure if I
No,
wanted
it looks fine to me. It looks good.
Good.
Okay, great. Okay, so I guess we'll get going. So welcome back, everyone. It's my great pleasure to introduce our next speaker, Dr. Suzanne Appleyard. Dr. Appleyard completed her bachelor's in pharmacology at the University College London. She then went on to complete a PhD in pharmacology and neurobiology at the University of Washington with Charles Chapkin, where she studied regulation of the kappa opioid receptor. She was a postdoc fellow at OHSU in Michael Anderson's lab, studying neuronal populations within the brainstem nucleus of the solitary tract, which are activated by visceral afferents. She's been at Washington State University since 2007. Dr. Appleyard's research focuses on energy homeostasis, NTS function and circuitry, and leptin signal transduction mechanisms using mouse genetic, electrophysiological, and behavioral approaches. Today, Dr. Appleyard will share with us her work entitled Modulation of Vagal Synaptic Transmission, Changing How the Gut Talks to the Brain.
Great, thank you, Lindsay. And I want to start by thanking Lindsay for organizing this. It is a great pleasure to see people and talk to people, even if it is online. It feels it's very nice to do that, given the isolation that we have been in. And I want to start by acknowledging the people who did the work. I've tried to put their picture at some point in the slide to acknowledge exactly who has done the work. And obviously also none of this would be possible without funding support from NIH, as well as from WSU and an alcohol and drug abuse research program here in the state of Washington. So I've had a very much a privilege to be able to follow the great introduction of the vagus. Okay. And so I really don't need to outline too much what the role is. But as Ivan was mentioning, the vagus nerve carries information coming from the gastrointestinal system, and it sends that message back up into the brain. And it terminates in the nucleus of the solitary tract in the brainstem. And then neurons that are located within the NTS send projections throughout the rest of the brain, including the parabrachial nucleus that we just heard about, also to areas within the hypothalamus, as well as other forebrain regions, and also locally back within the brainstem. And one thing that has always fascinated me is that all of this rich information that is being carried by the incoming vagus about the state of the viscera impacts these MTS neurons that then send it straight away throughout the rest of the brain. And many of these MTS neurons actually can bifurcate or even trifurcate so that the information can be sent in a coordinated manner to multiple brain regions. And this means that this one synapse that's located within the NTS will have a big impact on the information that is coming from the periphery and then what is sent on to the rest of the brain. And one other thing that's interesting about the nucleus of the solitary tract is it's located, sorry, I meant to say that when the signal goes on to the rest of the brain, as Ivan was talking about, this can send the feeling of satiation, which isn't always a negative association, but can have some positive affect related to it. And as Ivan was talking about, we will, sorry, I apologize. I'm having trouble with my computer. We have to add on the fact that this information now is also carrying positive valence about the presence of food in the stomach. But one very interesting thing I find about the NTS is it's located right beneath an area, the area postrema in the brainstem, which is a circumventricular organ that contains a lot of blood vessels. And indeed, some parts of the NTS around the border of the AP may also contain fenestrated capillaries, and therefore be able to potentially sense circulating hormones and potentially blood glucose. In addition, the NTS receives a lot of information from the rest of the brain about many different aspects related to food intake. And these are carried by multiple different neurotransmitters that have the potential to impact transmission across this vital synapse. And so one question we are really interested in is how these NTS neurons integrate information that's coming in from the vagus nerve with other information carried by hormones and neurotransmitters and metabolic state to impact transmission to downstream targets. So the way we answer this question is to take advantage of the anatomy of the vagus nerve, which is when it enters into the brain, it actually then runs from rostral to caudal along the fourth ventricle. So this means we can take quasi-horizontal slices that will contain the incoming vagal afferents in the solitary tract. And you can see one such slice here. You've got a quasi-horizontal slice demonstrated here. And can I just check, you can see the arrow that I am using, correct?
Actually, no. Oh, yes, it's very tiny.
Okay. This quasi-horizontal slice contains large amounts of the solitary tract in place. And this allows us to selectively activate those incoming vagal afferents as shown with the yellow arrow. And then we can record the effect of the release of the transmitter in the NTS neurons. We actually take advantage, we use both the rat and the mouse models, and we use wholesale patch clamp techniques in order to record the responses to vagal activation. We have been using mice a lot recently, and that is to take advantage of the many different transgenic lines that allow us to identify specific populations of NTS neurons. Because the NTS neurons themselves have a very varied transmitter phenotype. In addition to the classic transmitters, glutamate and GABA, there's a lot of peptides that are expressed within the NTS. And so it's a very heterogeneous population of neurons. And by using existing tools, such as the transgenic mouse that expresses enhanced green fluorescent protein under the control of the tyrosine hydroxylase promoter, we can identify specific populations. In this case, the population of catecholamine neurons that will include dopaminergic, norepinephrine or noradrenergic, and epinephrine neurons. In the region of the NTS that we record from, the majority are thought to be noradrenergic or norepinephrine neurons. And I should mention, because we have talked about, we've heard from the previous speakers, about the different sections of the NTS. And if you'll note, the region that we record in is further caudal NTS. And this is the region that Aban was talking about that receives many of those inputs from the gastrointestinal system. The inputs regarding taste tend to come in more rostrally. But here we are looking at the ability of the vagus to impact caudal NTS neurons. And we can actually specifically label populations of neurons from the stomach, for example, with DI, and show that up here in the top, this is a gastric label where we put DI into the stomach. And you can see that there are the nerve terminals that contain the gastric afferents making connections with an NTS catecholamine neuron that we can then record from. We do use other mouse models that I'll talk about today. Is one that is where the promoter for the leptin receptor is driving Cree, and we cross that with the Rosa Lox-P tomato mouse that was made by the Allen Brain Institute in order to identify neurons that specifically express the leptin receptor. And I also want to note at this stage that we do use both male and female animals. We do keep track of which sex the response is in. However, to date, we really have not seen a large sex difference. That is with the understanding that the number that we're using, the power that we're using with our experiments is unlikely to detect a subtle sex difference. But we haven't seen a big difference between male and female animals in the data that I'm going to talk about today. And as age can also make a difference, these animals are recorded from usually between 6 and 16 weeks old. So, I apologize, my mouse is causing trouble. So why do we specifically focus on the catecholamine neurons themselves to begin with when we first started these studies? And that is for several reasons. There had been a lot of different groups that had shown that these tyrosine hydroxylase neurons located in the NTS are activated at the level of gene transcription following ingestion of a meal, and also stimuli that are associated with ingestion of meals, such as gastric distension, cholecystokinin, leptin, free fatty acids, and amino acids. Linda Reinemann also had shown that ablation of these NTS catecholamine neurons using a saporotoxin approach results in reduced effects of CCK to inhibit food intake. More recently, we also showed that these neurons do lie directly downstream of vagal afferent, identifying them as a population that are capable of responding to these incoming vagal afferent stimuli. And then more recently, Richard Parmiter's group have shown that either using DREADS or optogenetics, activation of this population using DREADS driven by dopamine beta hydroxylase. So this will identify both noradrenergic and adrenergic neurons, but not the dopamine neurons. Activation of those neurons inhibits food intake. And they also went on to show that this included specifically a projection to parabrachial nucleus. Although even within one population that's identified with a promoter, there is heterogeneity. A recent group just showed, in this case, using tyrosine hydroxylase promoter to drive channel rhodopsin, showed that the projection up to the arcuate nucleus actually stimulates food intake, which is the opposite to what had been seen previously, suggesting there is some heterogeneity in this population. So what do we see when we take this horizontal slice and we stimulate the vagus? This shows you a typical response that we will see. Here is an artifact, which I hope you can see with the arrow. And then shortly after that, there is a large excitatory postsynaptic current. This is multiple stimulations in a row. And I think you can see that the onset or latency to the start of the current is very consistent, as is the size and shape of the current. All of these properties indicate that it's a direct or monosynaptic connection from the vagal afferent itself straight onto the catecholamine neuron. And we believe it reflects release of glutamate from vagal afferent terminals that then binds to AMPA receptors, as this can be blocked by antagonist of the AMPA receptor, which results in EPSC that I showed you, and then generation of an action potential. And for any information coming in from the vagus to be transmitted to downstream targets at different brain regions, especially those obviously a long way away, an action potential has to be generated. And then ultimately that leads to the signal that I have had enough to eat, or as Yvonne was just talking about, that food was good. And as I mentioned, one thing that struck me about this is that this one synapse is really critical for determining the size and degree of information that's coming from the vagus that's then transmitted throughout the brain. So how can this synaptic transmission be altered? And we've shown it can be altered in several different ways. The first approach we looked at was to look at the effects of orexigenic compounds. Those are compounds that stimulate food intake. And here I'm showing you the effects of methencephalin, which is an endogenous opioid. And opioids are known to stimulate food intake, including at the level of the NTS. And you can see the bath application of methencephalin significantly inhibits the size of the excitatory postsynaptic current that we see in the catecholamine neuron. And using pharmacology, we showed, consistent with the behavioral effects, that this effect is mediated through the mu-opioid receptor. In addition, another compound that is known to stimulate food intake, MPY, also has a very similar effect. If you look at the size of the first EPSC, in the presence of MPY, this EPSC is greatly reduced. And again, using pharmacology, we can show that this was through activation of Y2 receptors on the vagal afferent terminals. And again, this is consistent with recent behavioral paper by Bob Rich's lab showing that Y2 agonists seem to mediate the stimulation of food intake seen with MPY centrally. In addition, when we look at the hormone ghrelin, which is a hormone that is whose levels rise prior to a meal and is thought to be important for stimulating food intake, in the presence of ghrelin, again, this ability of the vagus to activate these NTS neurons is greatly reduced. And this appears to be through the GHSR receptor. So all of these compounds can act presynaptically to decrease the amount of glutamate that's released, decrease the size of the EPSC. But an important question is the only way that the NTS neuron or cell body can talk to its downstream targets is by generation of an action potential. So a critical question is, does this actually impact the ability of NTS neurons to generate an action potential in response to the vagus? And here we're recording in current clamps. So now you're looking at depolarizations and then firing of action potentials. And each of the black triangles is stimulation, an individual stimulation of the solitary tract containing the vagal afferents. Under control conditions, you can see each stimulation of the vagus has a high fidelity to produce an action potential in the NTS catecholamine neurons. In the presence of MPY, this ability is greatly reduced. And if you look at throughput, under control conditions, greater than 80% of the time, stimulation of the vagus results in an action potential, but it drops down to less than 50% in the presence of MPY. We see similar effects with metenkephalin, in fact, consistent with metenkephalin's dramatic effect to decrease the size of the evoked EPSC, is very rare to see an action potential being generated in the presence of metenkephalin. It seems to really inhibit the ability of vagus to activate NTS neurons. So this suggests that there will be less action potential firing, and potentially it means that the rest of the brain won't receive the signals from the vagus and might contribute to increased meal size, which all of these compounds produce, as the rat is feeling, I'm not quite full yet. One thing that we noticed when we were looking at the ability of these orexigenic compounds to inhibit vagal activation is that in general, you get a very large, if you just look at the spontaneous EPSCs that you record in these neurons, they are quite large and they are quite frequent. And we were wondering, given the size and frequency of these excitatory events, is it possible that you can drive the activity of NTS neurons independent of vagal firing? So we are all very well aware of the normal dog, when the vagus fires, the NTS responds, and then the information is passed on. But this would mean that it would, NTS neurons could be driven in the absence of vagal firing. And when we looked at this, we looked at a couple of different compounds. And we started with serotonin. As we heard about in the previous talk, serotonin is highly expressed in the gut, and it will drive firing of vagal afferents from the periphery to the center. But the 5-HT3 receptors are also found within the NTS on those vagal afferent terminals. And so we bath applied the serotonin, and you can see that in the presence of serotonin, these so-called spontaneous glutamate events, their frequency is significantly increased, and this effect over time can be shown in the bottom graph. So serotonin significantly drives the release of glutamate, and we did several experiments to show that, suggest that it is coming from the vagal afferent terminals, but it's being released in the absence of stimulation of the vagus itself. And again, we use pharmacology to confirm that this is through activation of the 5-HT3 receptor. That's the ligand-gated ion channel serotonin receptor. And another compound that we looked at was nicotine, as I'm sure people are very aware that nicotine is another compound that can significantly reduce food intake. It has very broad actions throughout the brain. But when you look at its effects on NTS neurons itself, it also it causes an increase in glutamate release. In this case, we are puffing the drug, and that is because nicotinic receptors can very quickly desensitize or inactivate. And so we don't bath apply drugs, we puff them to allow activation of those receptors and then recovery before we puff again. In this case, we're giving an agonist at the alpha-7 nicotinic receptor, and you can see a dramatic increase in glutamatergic events. And then lastly, the one I'm showing you is the effect of CCK itself. Again, CCK is found in the periphery. It is known to activate vagal afferent terminals in the periphery. But the CCK receptors are also found on vagal afferent terminals within the NTS. And when you bath apply CCK, you do get this dramatic increase in glutamate release and an increase in the frequency of these excitatory events. Again, these are just showing that there's an increase in glutamate release, and that is capable of activating excitatory postsynaptic currents in these NTS neurons. But ultimately, the question is this sufficient to generate action potentials? Because again, the way the NTS neurons communicate with their downstream targets is by generation of an action potential. So to look at this, we again use current clamp approach. But here we're just looking at basal firing within a neuron. And in this case, we're bath applying a 5-HT3 receptor agonist. And you can see after the delay for the perfusion that the 5-HT3 receptor agonist causes an increase in firing rate, and that is quantified here. If this is truly due to an increase in glutamate from these vagal afferent terminals, you would expect that any effect would be blocked if you blocked the glutamate receptors themselves, because then the NTS neurons will not be able to respond to the glutamate and would not be able to alter their firing in response. And that's exactly what we see in the presence of the AMPA receptor antagonist, NBQX. We see that there is now the 5-HT3 receptor agonist no longer is capable of driving increased firing in these neurons, confirming that it is through increase in glutamate release. And we see very similar effects when we look at the alpha-7 nicotinic agonist. Again, the agonist alone is capable of driving firing within these neurons, but in the presence of the glutamate receptor antagonist, this agonist can no longer drive the activity of these neurons, suggesting that it is through increasing glutamate release from these terminals that is then causing NTS neurons to fire. And one thing, again, that I find very interesting with this is it opens up the possibility, at least, that you could elicit feelings similar to what happens when you stimulate the vagus, but through directly activating NTS neurons. So a rat might feel or a mouse might feel satiated, even in the absence of having eaten, if you get release of serotonin or acetylcholine locally. But ultimately, that is the question, right? We're showing that CCK, serotonin, and nicotine or acetylcholine are all capable of driving release of glutamate from the vagus and driving NTS neurons. But these are exogenously applied. And so it could just be the receptor happens to have been sent both to the periphery and to the center. And so you're just pharmacologically identifying the types of vagal inputs that these neurons receive. And so we really are interested in looking at what is the endogenous regulation. Is this a physiologically important pathway by which NTS neuronal activity could be controlled and, as it were, usurp the vagus or take over the vagus? And so today I'm just going to show you one example, and it's by graduate student Stephen Page. And what he did was to cross mice that expressed channel rhodopsin under the control of the choline acetyltransferase, or CHAT promoter, which will mean that channel rhodopsin is expressed in cholinergic fibers. And then he then crossed these mice with our TH-EGFP mice. And now you have a mouse where you can identify the presence of the the catecholamine neuron and record specifically in catecholamine neurons while stimulating the release of acetylcholine. When Steve did this, what he saw is that you, with light activation, you do get this transient increase in glutamate events. So it does look like that light activation is causing an increase in glutamate from onto the NTS catecholamine neuron. And it's not seen if the channel rhodopsin is not there. It is not some non-specific effect of the light itself. It's quantified down in the graph on the bottom left. And Steve went on to show that this is also through the alpha-7, so it really is through release of acetylcholine that is binding to alpha-7 receptors and driving the release, because if you bath apply the alpha-7 receptor antagonist, then you no longer see this effect. So again, is that sufficient to drive action potentials in these neurons? And the answer is yes. You can see again, with the light activation, you get a transient increase in the frequency of action potential firing. So this represents a mechanism, and I emphasize it represents a mechanism whereby these compounds can potentially drive NTS neuronal activity. What about the flip? Can compounds that we know actually tend to promote food intake, do they actually have effect on this spontaneous glutamate? And again, we've looked at ghrelin, metenkephalin, and MTY, but what I'm going to show you today is the effects of ghrelin. And ghrelin, when ghrelin is bath applied, you can see that the frequency of the spontaneous EPSCs is reduced. And this does actually translate into a decrease in action potential firing. So when ghrelin is around, the neuron decreases its firing, and once it washes out, firing is restored. Interestingly, you can also block the effects of ghrelin with MBQX, the glutamate receptor antagonist, suggesting it's not that ghrelin is having a direct effect on the NTS neurons, but it is altering their activity by altering glutamate release from vagal afferent terminals. So what I've shown you so far is the ability of the vagal afferent terminal itself to be able to respond to multiple inputs, both ones that are known to promote more food intake, that act to decrease vagal activation of NTS neurons, as well as ones that are thought to inhibit food intake, that seem to be able to increase glutamate release, even independent of vagal afferent firing, and drive the activity of NTS neurons. But one question is, are these responsive plastic? That is, can they be altered by metabolic state? And we've looked at a couple of different metabolic states to see how this influences this synaptic transmission. And first, I'm showing you the effect of glucose. And if you remember at the beginning, I said that the NTS lies below the area of the strema and potentially has some fenestrated capillaries. I would love to know what the actual concentration of glucose is within the NTS. It is a question I would really, really like to know. Obviously, if it is sensing blood glucose, then it is possible that the glucose levels would be getting up to the levels that you see in the blood, which would be, you know, could potentially get up to the 9 or 10 millimolar levels. And so in order to look at the effects of glucose, we actually looked over a very wide glucose concentration because of this reason that we're not 100% certain what the actual concentration of glucose is in the NTS. And so we compared 10, 5, 2, and 1 millimolar. And here I'm just showing you the 5 and the 2 millimolar. And SR is again the 5-HT3 receptor agonist that drives glutamate release and drives firing. And it's expressed here as a fold change in spontaneous EPSC frequency. So in 5 millimolar glucose, in the presence of SR, you have about a 5-fold increase in the amount of glutamate, spontaneous glutamate events. And the gray is the wash. But you can see in 2 millimolar, now SR only has a very subtle, at best, effect to increase glutamate release. So it's quite a large difference from 5 millimolar to 2 millimolar in the amount of excitation that serotonin can have on these vagal afferent terminals. And we see very similar effects when we look at acetylcholine. In this case, we're using the chat channel rhodopsin mice crossed with a THEGFP to be able to specifically photostimulate release of acetylcholine and look at its ability to increase the spontaneous glutamate events or spontaneous EPSCs. Here I'm showing you the difference from 10 to 2, which is a much larger difference. But again, at the higher glucose levels, you're getting a large increase in the number of glutamate events that the NTS neurons see. But when you drop to 2 millimolar, the size of this effect goes down. And this would suggest that under conditions of high glucose, you're going to get much more activation of NTS neurons, which presumably would promote overall a feeling of satiation. Whereas in lower glucose conditions, which generally occur when an animal is low on nutrients, the effect is much less. And remembering that ultimately activation of these NTS catecholamine neurons in general tends to cause inhibition of food intake, the fact that you're decreasing a pathway that tends to inhibit food intake would allow, presumably, the animal to eat more. We also looked in fasted and fed conditions, and for this, I'm showing you the effects of ghrelin, which again, remember, is the hormone that circulates in the blood and rises before a meal. And ghrelin's effect in a non-fasted animal, it significantly inhibits the size of the evoked GPSC, but it's fairly subtle and quite variable from cell to cell. But in a fasted animal, ghrelin has a much stronger effect to inhibit vagal activation of NTS catecholamine neurons, which again suggests under fasted state and high levels, when there are higher levels of ghrelin, you also have this increased sensitivity to ghrelin that would tend to decrease the ability of the vagus to activate NTS catecholamine neurons, and presumably, again, would allow an animal to eat longer. So to this point, I've shown you the effects on a presynaptic terminal. So Ivan mentioned this before, the vagus is very good at being able to integrate a lot of different signals. And ultimately, how much glutamate release you get and how much activation you have of NTS neurons is going to depend on the levels of each of these compounds. But we were also interested, can you change the sensitivity or the gain of the system by changing the postsynaptic response? To do this, we again do patch clamp recordings from NTS neurons, and this work was done by Juan Zhao, a postdoc in the lab. And what Juan found is something that many labs who look at vagal activation of NTS neurons have seen, and that is that when you stimulate the vagus with 10 stimulations at 50 hertz, which following a meal, the vagus is thought to fire at quite high levels, anywhere from 10 to 50 hertz. And so this is within physiological range following a meal. What you see with the AMPA response, that's the one mediated by AMPA receptors, is that the first EPSC is very large, and then the amplitude decreases over time. And this is thought to be because this is a very high probability of release synapse. So when you stimulate the vagus, you get a very large release of glutamate, that then means when a second stimulation comes too soon, the terminal hasn't had a chance to restore those glutamate vesicles, and then not available for release. So you've got less vesicles available for release, less glutamate released, and therefore a smaller effect. And this frequency-dependent depression, as I said, has been well documented by people reporting in the NTS. But in addition to AMPA receptors, there are also NMDA receptors found within the NTS and including within the NTS neurons themselves. And when you record, for those, when you're thinking about the NMDA receptor, one of its classic properties is that under hyperpolarized conditions, there is a magnesium block, which you need to depolarize a neuron in order to remove. And if you do record under conditions where you remove that magnesium block and then you stimulate the vagus, what you will see is this additional slow decaying component of an EPSC. In fact, it decays so slowly that it hasn't actually gone back to baseline before the second stimulation. So the result is you get a sustained current. And this can be blocked by NMDA receptor antagonists. So it is mediated by NMDA. And it is due to two main properties. One is that slower decay of the current, but also NMDA receptors actually have a higher affinity for glutamate than AMPA. It is frequency dependent, so the higher the frequency you drive the vagus at, the larger the size of your NMDA current. And ultimately, again, the question is, does this affect generation of action potentials? And this is showing you a prolonged stimulation, 100 stimulations of the vagus. And you can see that during that time point, you get this large depolarization and continued action potential firing in the NTS neuron. In the presence of the NMDA antagonist now, you decrease the frequency of this firing, suggesting that NMDAs are important for the fidelity of transmission across this synapse. And this is consistent with behavioral work that's been known for a while out of Bob Ritter's lab, where they showed that CCK normally inhibits food intake, but if you have an NMDA receptor antagonist around, this effect is lost, suggesting that NMDA receptors are very important for this phenomenon. And this may well be one mechanism underlying that. And NMDA receptors are fascinating because their activity is highly able to be modulated. And one thing we were interested in is leptin's ability to modulate it. And that's because leptin receptors are also found on vagal afferent terminals in the NTS and on NTS neurons. And leptin is known to enhance or increase the sensitivity of the NTS to gastric nutrients and CCK. Leptin injection straight into the NTS will reduce food intake. And here we're particularly examining the role of a postsynaptic leptin receptor. And that's because knockdown of those postsynaptic receptors does result in hyperphagia and weight gain. And in relation to the NMDA, it's known that leptin can enhance NMDA receptors in the hippocampus. And so the suggestion is that leptin might be able to potentiate the size of the NMDA current and increase the sensitivity of NTS neurons further. Also, Martin Meyer's group recently showed that stimulation of these leptin receptor neurons is also capable of causing inhibition of food intake. So this is work by Drew Nyans, who just graduated and got his PhD. And what Drew found is that when he bath applies leptin, the size of this NMDA current is increased. And you can see this occurring over time. It takes about 10 to 15 minutes for the maximal effect of leptin to occur. And this shows you the distribution responses across neurons. And again, these are both neurons from male and female mice. Again, when you look at the effect of leptin on throughput, here Drew is stimulating the vagus 20 times. And he is doing it with setting the throughput at about 30%, and leptin will increase this. And that again is shown here. And this effect of leptin to increase throughput is blocked by NMDA receptor antagonists. So to summarize what I've shown you, that the GI feedback is capable of, vagal activation is capable of releasing glutamate in the NTS. It drives NTS neurons, which then has effects throughout the brain. Leptin seems to be able to increase this NMDA and potentiate the feeling of the rat being full. And indeed, studies from Bob and Than have shown that leptin inhibits food intake when given at the level of the NTS, but it fails to do so in the presence of NMDA receptor antagonists.
So actually, Susanna, we're running a little bit.
I know, I noticed that, and I apologize. I'm gonna go to the end, and I had this timed at exactly 30, so I'm confused. Anyway, basically, to summarize all of the things that I've shown you, this synapse is very important and is capable of being modulated by multiple different signals that can impact what the rest of the brain is seeing. And I will just point out to people to bring it all together with other people's. I'm doing one model synapse, but there's lots of different populations of NTS neurons, lots of different phenotypes of afferents and hearing. And the question is really going to be, how are these pathways all influenced by all these things to produce the behavioral effect at the end? Oops. Thank you again for your attention, and I apologize for going long.
Sorry, I didn't realize you're almost at the end there. Is what I wasn't quite sure.
I apologize. I think so.
I know I ran long myself, so I'm also to blame. Thank you very much, Suzanne. So we'll continue straight away with Dr. Lindsay Shear, who is here to talk about her work. Dr. Shear received her PhD at Purdue University, working with Terry Powley to investigate the effects of taste stimuli within the digestive tract on ingestive behaviors. As a postdoc at Florida State University with Dr. Alan Spector, Dr. Shear continued to investigate this role in taste and in taste receptor mediated and taste receptor independent signaling on modulating feeding behaviors. Now, as an assistant professor at the University of Southern California, Dr. Shear's lab investigates how the chemical constituents of food and fluids are sensed and how these oral and post-oral signals are processed in the brain. Today, she'll share her recent work with a talk entitled Oral and Post-Oral Sensors Linked to Glucose Appetite. Thanks for joining us, Lindsay.
Hey, good afternoon. Greetings from Los Angeles, everyone. I want to just start by thanking Lindsay for organizing this and inviting me to join this. It's been a really excellent symposium today. I'm going to round out things today by presenting three main lines of emerging work from my lab, centered around a distributed network of sensors and integrators that are really involved in promoting appetite for very specific sugar glucose. So we've known for some time that obesity and diabetes which are now among the leading causes of preventable death in the US, are linked to the chronic overconsumption of simple sugars. And for many years, the proverbial sweet tooth has really shouldered the blame for this. And that's because a variety of sugars that you would normally see in the diet, as well as artificial sweeteners, bind to a single taste receptor in the periphery. And Alan talked about this earlier today. The T1R2 plus 3 heterodimeric G protein coupled receptor. Activation of this receptor ultimately stimulates release of dopamine in reward areas of the brain, inherently elicits a pleasurable sensation in humans, and really drives consumption in humans and other animals. However, we know that not all sweeteners, even though they bind to this single taste receptor, are created equal, and some sweet compounds yield little to no calories, those artificial sweeteners I mentioned. But even among the caloric sugars, there are a diversity of metabolic fates for these sugars. And these differences in terms of how they influence intake and preference are really apparent practically as soon as they bypass or pass those taste receptors and hit the gut. And we showed that in a very simple experiment that I did in Alan Spector's lab when I was a postdoc, in which we gave animals a single bottle of a sugar to drink and measured their licking across a 30-minute meal test session. And they were given equicaloric solutions of either glucose or fructose to drink. And I'll first just start out by showing you what I'm highlighting in the yellow outline panel here is that in the very first minute of the session, intake was identical for these two sugars. And that, again, is consistent with this model that they're activating these taste receptors and driving consumption from there in a very similar manner. But within just a minute or two after that, we saw this really robust amplification of lick rate in the animals that were given the glucose to consume relative to those that were given fructose to consume. And so this sort of provided an additional line of evidence that glucose is really a more potent reinforcer or stimulator of intake than ecocaloric fructose. And the sort of time course with which we were seeing this suggested that it was probably happening based on a signal that was coming from the gut and very likely a neural signal. And I'll talk a little bit more about that in a few slides. And if you, sorry, I don't know why if you let animals have a choice between these two sugars over a more extended period of time across several meals, in fact, across 48 hours, what we see and what others have shown as well is that they very strongly prefer the glucose over the fructose. Even though these two sugars would ultimately provide the same number of calories, they almost exclusively drink the glucose. And so this has really taken, along with other evidence that's been accumulating over the last 30 to 40 years, that glucose is a really potent reinforcer of ingestion. It also conditions strong preferences for cues like flavors and locations, and this is likely happening from post-ingestive site of action. But in terms of, I just want to kind of take a couple of minutes here to discuss potentially where these signals are coming from. Well, it's clear, at least from the behavioral data, and I'll bring up this, remind you of this. Oh, there's a delay. There we go. Bring you up this slide that I showed earlier. Well, it's clear that just based on the temporal dynamics that these initial signals are coming from the gut. It's very likely that glucose is, in fact, we have evidence from other studies that glucose is activating and stimulating and reinforcing these preferences from other sites. And so even within the course of a meal, these sugars are going to hit receptors from the tongue to the gut, as well as release hormones that are going to get into circulation, as well as serve as signals themselves once they get into circulation. And so trying to sort of figure out and parse out which signals are involved in driving this potent glucose appetite is sort of difficult, because there's a cascade of signals that this is going to generate within a course of a meal, and of course that in the hours that follow the meal. But we, like others, have really started by zeroing in on the vagus nerve, which you've already heard quite a bit about today. Because of that, the really robust and rapid response that we were seeing, we figured that this is probably involving the vagus nerve, which is the most well-studied gut-brain route of communication. And so what we did to look at whether or not this nerve was required for this rapid glucose appetition that we were seeing is to kind of take a sledgehammer approach. And what we did was perform some bilateral subdiaphragmatic vagotomies in our rats, in which we severed the vagus nerve just below the diaphragm. So that's going to cut off communication from the GI organs to the brain. And we then did a series of tests, which I'll outline here. We did our single 30-minute meal-like tests in which the animals were given glucose or fructose to consume. And then we followed this up with a long-term 48-hour 2-bottle choice test. And all of this to assess whether or not the vagus nerve was required for both the immediate glucose appetition effect and then that long-term preference for glucose. And I want to take a second to just point out that we're not the first ones to do this. There have been a number of different studies that have tried to sort of interrogate the role of the vagus nerve in nutrient-related appetite, specifically with sugars and fats. And at least when it comes to sugar, this has been a little bit elusive. It's been sort of hard to nail down a necessary role for the vagus, and I think this is and stemming from a couple of different factors. One possibility being that, you know, the various methods that are available to lesion the vagus are sometimes incomplete, and so any spared fibers might be sufficient to carry the signal. And then also, as I said, there seems to be this kind of redundancy of signals that the animals could pick up on throughout the course of a meal and the hours after a meal that would be sufficient to promote appetite. And so we're sort of well aware of all of the studies that have tried to track this down and the difficulties associated with them. So that's the reason we took this sledgehammer approach. So I'm going to start with the sort of the end of the story here and present the 24-hour preference test data, because these would not be surprising at all. We found that animals that were given the opportunity to sample between glucose and fructose, as of course, as you would expect in the shams, and I've already shown, they highly prefer the glucose relative to fructose, but we saw a very similar preference in our SDVs. So wiping out that vagal connection between the gut and the brain did not impair their ability to learn to prefer the glucose over the fructose. Again, that's not surprising just based on all of the other studies that have attempted to look at this. But when we backtracked and looked at those early meal sessions and looked at that rapid glucose appetition phase, in a really high resolution manner where we could track minute by minute lick rate, what we found, again, these are our sham animals, but we saw that very typical early meal glucose appetition effect in the sham animals that I'd shown you before. And what we saw in our SDV animals was that early phase appetition was completely eliminated by the SDV. And so that suggested to us that the vagal gut-brain signaling is required for that rapid glucose appetition. But again, as others have shown, it's really not critical for the development of preference for glucose over, in our case, fructose, but others have shown it with other sweeteners and water over longer periods. So we then went on to look at what, once these signals are arising into the brain from the gut by the vagus, potentially what are the central brain regions that are involved in processing this very glucose-specific appetite. And I don't have time to go into all the details on this now, but we did a sort of a brain-wide mapping of areas that are responding differentially to glucose and fructose, calorie-matched solutions of those in the areas that are responding to those two different sugars in a different way. And we identified a number of different regions this way. And we limited these to neurons that were, or neuron populations that were responding to very short-term intake to kind of capture that rapid early meal appetition effect that we see that the vagus is involved in. And so we were zeroed in on the arcuate nucleus for a number of different reasons. One being that it was identified in this brain-wide mapping study that we did. Another is that this area is highly enriched with glucose sensors, and those glucose sensors express this enzyme glucokinase, which is a marker of these sensors. But essentially, what it does is link extracellular glucose levels to neuronal output in cells. And another key reason that we zeroed in on the arcuate nucleus, the hypothalamus, is this study that came out in 2015 by Hussein et al. That artificially, virally activated glucokinase in these glucosensory cells in the arcuate, and then measured glucose versus fructose intake over a 24-hour period. And what they found was that the animals that had increased glucokinase activity selectively consumed more glucose, and there was no effect on fructose consumption. And so this sort of paralleled or mirrored what we were seeing in our short-term intake sessions, that there was a system that was promoting glucose intake without an effect on fructose intake. And so another reason we zeroed in on that. But again, this study was happening, the This intake was measured over a 24-hour period, so there's a lot of different events that are going on related to glucose and glucose metabolism during that time. And so we wanted to get at whether or not this region, in particular, these kinase expressing neurons in the arcuate, were involved in that rapid short-term meal-like appetitious effect of glucose. And so the first pass at this was, and now I'm going to present some new work from the lab. First pass at this was to just look to see whether or not glucokinase was upregulated in the arcuate nucleus in response to short-term rapid consumption of sugar. And we did this by giving rats one of 3 different types of sugar: glucose, calorie-matched fructose, or alpha-MDG, which is a non-metabolizable glucose analog. We gave them, they just had a few minutes to consume it. You can see the intake here was matched very well over a short duration. The control group didn't have any solution to drink, so they were just kept under the same conditions, but otherwise didn't have any solution to drink during that time. And then we, at the end of that time, we harvested the mediobasal hypothalamus contained in the arcuate, and we looked at glucokinase expression using qPCR. And what I'm showing here is we found that glucose rapidly upregulated glucokinase expression in the arcuate, more so than the other sugars, and in fact, those other sugars were no different than the control condition. And so we saw the selective driving of glucokinase expression in the arcuate by short-term consumption of glucose. So then the next step to this was then to see whether or not we could artificially, pharmacologically activate glucokinase in the arcuate, and then similarly drive intake, as Hussein et al. Did over 24 hours, but whether or not we would drive that intake within a short meal-like session. And so we used we implanted cannula directly into the arcuate and delivered a glucokinase activating compound known as compound A directly before a 30-minute session in which the animals were given glucose or fructose to drink. And what we found here is that animals that had glucokinase activation consumed significantly more glucose than those that were just given artificial cerebrospinal fluid control. And we had no effect of the compound A on fructose intake. And I should mention these were ad libitum fed animals, so they were not necessarily in a deprived state or in need of glucose. So this was amplification of intake even in an ad lib animal. So activation of glucokinase in the arcuate specifically drives short-term glucose intake. We then wanted to sort of look at whether or not glucokinase in the arcuate was required for this thin meal appetition effect. And so we did this by taking advantage of a custom designed virus in which we had a short hairpin RNA to knock down glucokinase expression in the arcuate directly, so stereotaxically delivered into the arcuate. And what we saw was that animals that had this selective knockdown of glucokinase in the arcuate nucleus consumed pretty similar amounts of glucose and fructose in a short-term meal test. And in fact, when we looked at, took a little bit closer look and looked at their minute-by-minute licking patterns, basically completely abolished that early phase glucose appetition effect when we knocked down glucokinase in the arcuate nucleus. So just to kind of provide an interim summary of what I've said so far, within a meal, glucose produces this rapid post-ingestive effect that in turn promotes overconsumption. The vagus nerve is required for this immediate appetitious effect, though again, as others have shown, other inputs may be involved and are certainly sufficient to promote glucose ingestion over longer periods of time. And glucose-sensitive neurons expressing glucokinase in the arcuate nucleus of the hypothalamus appear to play a role in the central control of this rapid within-meal glucose appetite. Okay, so I want to circle back to the gustatory system. I dismissed it a little bit at the beginning of the talk, and all for setup for this point here, but started to kind of look at thinking about what's happening over the long term when animals have extended access to sugars that have very different metabolic fates, and they can clearly figure out which is the glucose from the fructose in this case, and use that to their advantage to get more glucose. So we started to kind of question whether or not having this single taste receptor that basically treats all of the sugars and sweeteners the same was really adaptive, and whether or not potentially this experience with these two different, very different metabolically distinct sugars would actually sort of reprogram the way that the way that the taste system is responding to glucose. In other words, we looked to see whether or not there was potentially a hidden taste mechanism that would allow the animals to detect glucose-rich food sources in their environment, and whether or not that was really unmasked through experience with figuring out which sugars are providing the glucose. And so in order to begin to get at this is work that I started out when I was a postdoc in Alan Spector's lab, but we arranged a pretty straightforward and simple paradigm. Shifting over to mice now, but basically what we did is we had several different groups that had various different types of exposure to glucose in their diet, and we had one group, which we refer to as GBF, that had exposure to glucose and fructose over a 30-day period in which they were given 3 concentrations of each sugar in a randomized order. We used 3 different concentrations just so we could render any potential intensity differences in the sugars irrelevant to these animals. And so they had this sort of random exposure repeated over several weeks. We had a comparison group that had the same number of exposures, but they were only given glucose to consume during that time, so they're not getting any sort of they're not seeing any potential differences in metabolic effects of the sugars, just having experienced one sugar during that time. And then we had a third group that we kept naive to sugar and sweet taste altogether. We gave them calorie-matched corn oil and emulsions during this same period of time. And then the big test was at the end, in which we examined how they responded to the orosensory properties of these 2 different sugars in a brief access taste test. Now, before I get to that, I want to highlight that we included T1R2 and 3 knockout mice in addition to wild type controls in the study, because we were also looking at whether or not anything that, whether or not the T1R2 plus 3 receptor would be required to change any responsivity to these 2 different sugars as a function of this exposure. So in order to assess whether or not this exposure changed the way, this exposure to the 2 sugars changed the way the animals were responding to orosensory properties of the sugar, we ran a brief access taste test. And I'll just play this video. In this test, the animals get very brief 10-second presentations of sugar, and we give them mixed trials of glucose and fructose in a randomized order. And they have 30 minutes. They can initiate as many of these trials as they want. We measure licking responses during that time. And the nice feature of this test is that they get very small volumes in a randomized order, which really precludes any post-ingestive influences of the sugars from affecting their licking behavior. So it allows us to determine how they're responding to the taste properties of these sugars. So to get to these data, I'll show you first the naive group. So these are the animals that are just kept sugar naive, never had experienced sugar prior to this point. And what I'm plotting here is lick score across the 3 concentrations of each sugar. You could see the wild type animals respond just as you would expect. They lick to both sugars in a pretty comparable fashion, increasing as concentration increases. Again, this is exactly what you would expect if, based on a single receptor model, that these should be pretty similar to these mice. And the knockouts, you could see that that response to sugar is basically flatlined. I will point out there's a slight increase in licking to the glucose in the knockout mice, in the sugar naive knockout mice, though that didn't actually turn out to be statistically significant. They just basically wiped out concentration dependent responding for both sugars, and that's something that had been shown. So now to get to what happens when the animals have had extensive experience with glucose and fructose. Here I'm showing the wild type mice, and what you can see is a dramatically different pattern where these mice lick for glucose at a much higher level relative to fructose at each concentration that we tested. And so they're expressing that preference for glucose that we see in a 48-hour test here as an increase in licking responses to glucose over fructose at each concentration that we tested them. And what was interesting is that we saw the same exact pattern in the animals that had this canonical sweet taste receptor knocked out. So these animals lick, again, almost exclusively for glucose at all three concentrations tested. So there appears to be this experience-dependent increase in responding to the orosensory properties of glucose. So I wanted to kind of also show, I don't have time to go into all the details of this experiment, but we've done a follow-up experiment since in which we tested how these, not only how these animals respond to glucose and fructose, but other sugars that vary in their glucose content. And I'm just showing the snapshot of it here, but I wanted to show that this not only changes how these animals are, this type of sugar exposure not only changes how these animals are responding to orosensory properties of glucose, but they're also that's also translating into a change in how they respond to another disaccharide, glucose disaccharide maltose, where you see this heightened avidity for maltose relative to a sugar naive animal. And this is even up to the level of the highly preferred table sugar sucrose. And another point I want to make here is that this exposure doesn't kind of devalue sugar altogether or sweet taste altogether. In fact, these animals continue to respond very strongly to sucrose, but it's just that we're seeing an amplification and responding to other sugars that are not so intensely sweet and that are rich in glucose. And if we present just fructose to these sugar-exposed animals alone, they'll consume or lick avidly for fructose alone. So it seems that we're just sort of tacking on another family of compounds that they're highly motivated to consume. So we recognize a few different things about these patterns. First, that doesn't appear to require the canonical sweet taste receptor, and second, that it appears to be related to the glucose content of the sugar. And so we started to wonder whether or not there was a glucose sensor in the taste system that could be responsible for this. And so we looked, like others have, in a variety of other glucose sensors that are in other tissues, including the gut, pancreatic beta cell being probably the most well-studied model of glucose sensing cell. And of course, I've already talked about neurons. And indeed, a number of these glucose sensory elements have been, the ones I circled in ink there, had been found in taste cells before. This was a lot of work done by Bob Margolse's group and others. But whether or not those were linked to taste-guided behaviors, appetite, hadn't really been known. And we had we tested some of these options, and I don't have time to go into it, but we sort of realized that it's probably not SGLT1 that's required. So we focused on actually one that hadn't been recognized in the taste system before, and that is what I've already talked about, which is glucokinase, that we manipulated in the arcuate nucleus. And this is, as I mentioned, a key enzyme in translating glucose content into cellular output. And so we look to see whether or not sugar exposure, the type that I'm talking about, actually changes glucokinase in the taste buds. And what we did was we did our typical paradigm where we kept some animals naive, we gave some others this dual sugar exposure, and then we harvested their taste tissue and looked for glucokinase expression. And what we found is that glucokinase was upregulated in the taste tissue of sugar-exposed animals relative to sugar-naive animals. And so we thought this potentially could be a signaling mechanism through which the animals are able to detect glucose by the tongue, and that in turn gets translated into appetite. So we experimentally sort of tested this by taking sugar-naive mice and pharmacologically activating compound A in the taste tissue using compound A, the same drug that we use in arcuate, and measuring licking responses in a brief access test to glucose and fructose. And what I'm showing here is that we stimulate licking for glucose. These are we purposely chose very low concentrations of glucose, and so we amplify licking for glucose. We have no effect on licking for glucose when we activate glucokinase in the taste tissue. And we see a comparable response when we measure from the taste nerve, the chorda tympani nerve. Addition of glucokinase activator stimulates neural response to glucose, has no effect on matched concentrations of fructose. So activation of lingual glucokinase selectively stimulates licking for and taste nerve responsivity to glucose. So just to summarize these, there appears to be a previously unrecognized T1R2 plus 3 independent orosensory sugar receptor that's linked to appetite control. The motivational potency of the signals generated from this receptor are influenced by dietary experience. Glucokinase is expressed in murine taste buds. It's upregulated as a function of sugar exposure. I didn't show these data here, but we've also found it to be upregulated by periods of food restriction. And then local activation of this enzyme significantly bolsters licking and neural responsivity when glucose but not fructose is presented in the substrate. So this appears to be a reshaping of oral sensory inputs as a function of experience and feedback coming from gut and other sites about the nutrient content or the sort of the metabolic advantages of the food that's in the diet. And this is potentially an adaptive means through which intake can be titrated to need and even environmental availability at the earliest steps of the sensory processing. So now just to summarize all of that, the body's ability to locate, ingest, and officially assimilate glucose is key to health and survival. So there's perhaps no surprise then that the body employs a distributed network of glucose sensing mechanisms at key metabolic sites that successfully track glucose levels. And this is sort of a coordinated effort, and these signals are transmitted to central processors and even reshape peripheral sensory inputs to bias intake towards glucose-rich foods. So I'd like to just thank the people, and by thanking the people that did all of the hard work on these studies, and you for your attention, and I'm happy to take any questions.
Thanks so much, Lindsay. That was really a fascinating talk and really brought everything back around again. So we went from the tongue through the gut to the brain and then back out to the tongue again. So that's really is just how I imagine this symposium to go. So that's fantastic. Thanks so much. And do any of the panelists have a question for Lindsay while we're waiting for a few from the audience?
I have one if nobody is. So yeah, this is super interesting, this distinction between rapid versus more long-term, right? So when we did this type of vagotomy studies, we also noticed that they couldn't, they were not sufficient to abolish glucose preferences but they did to fats and amino acids like a glutamate, right? Even long term, the preference was totally gone. So I wonder if you have any ideas about, in terms of different macronutrients, how the rapid versus long-term signals play out. Why only for glucose the rapid preference learning seems to depend on the vagus, whereas for the others the whole learning seems to be impaired without the vagus. Um, do you have any ideas about that?
No, I mean it's something that we're interested in as well. And I've thought about just potentially just the redundancy in signals that are tracking for glucose might just sort of make it that more, or sort of reinforce detection of glucose. And I think these sensors might, one thing that we need to kind of sort out is whether or not these sensors are involved in, they're involved in appetite, but also glucose regulation, which is critical. And so just, I mean, I guess that's really been my only hypothesis about that, is that just key in controlling glucose levels and all that is just bolstered detectors of glucose in that way. And so really we're only capturing that very rapid, as soon as the glucose is hitting the gut, can wipe that out with a vagotomy. But as soon as it's getting absorbed and processed elsewhere, these other sensors are picking up on it.
Yeah, I'm interested in this because I have been pushing for the idea that part of the reward comes from a post-absorptive signal, but that signal does not seem to be relevant for other nutrients. So I'm really curious to hear, like, what people, those who are trying to parsing out the signal, think about it, right? And one thing that is true is that the glucose seems to be more reinforcing than any other nutrients if you do flavor conditioning on a caloric controlled manner. So yeah, probably there is a dual contribution from gut and post-absorptive.
Okay, well, I think we have scheduled a short break before we will reconvene for panel discussion. So please come back within the next 10 minutes. We'll reconvene at 4 p.m. Central Time, and we'll get going with our overall panel discussion. But to prepare, if any of the audience has questions they'd like to pose to the panelists, go ahead and type them into the chat at any time, or into the Q&A at any time, and then we'll kind of sort them out and see how they fit in to the discussion. Thanks very much, and I'll see you in a few minutes.
Hey guys, are you guys taking a break? Okay, go ahead, take a break. I hope I'm not going to echo throughout. Okay. So now we've seen a series of talks today on chemosensory signaling from the tongue and gut to the brain. Thank you for all staying with us. It looks like we've had a great big group today. It's really excellent. This is our first attempt at remote casting, and we really appreciate everyone's patience in enduring all of our technical difficulties. But this is the most fun part of the day, where we get to see everyone talk to one another and kind of say things that you wouldn't necessarily see in publication format, still somewhat on the record. So I want to go around the group and just have everyone quickly introduce themselves again and say hello, because this is going to be a standalone podcast and I want everyone's voice stamping here. So please, let's start with Susie. Oh.
Hi. Hi. Yeah, my name is Susie Appiot, and I'm a professor at Washington State University. And the main interest of my lab is to understand how NTS neurons process incoming vagal information before passing it on to the rest of the brain.
Great. Lindsay Shear, go ahead. I think I unmuted everyone.
Hi, I'm Lindsay Shear. I'm at the University of Southern California. My lab looks at nutrient detection along the GI tract from tongue to gut, and how that information is integrated centrally.
Yvonne.
Hi, I'm Ivan de Araujo. I am at Mount Sinai Hospital in New York at the Department of Neuroscience, and I work mostly on body-brain communication with an emphasis on how gut signals reach the central nervous system.
Sorry, I've been mispronouncing your name throughout.
Actually, there are so many pronunciations. I just picked one randomly.
Maya.
Hi, everyone. My name is Maya Kelber, and I'm faculty at Duke University, and I'm interested in sensory transduction in the gut.
Okay. And I think we just Lindsay McPherson.
Oh, there's Alan too.
Oh, we didn't do Alan. Alan, please.
Sorry.
Don't trust me with this.
I'm Alan Spector. I'm a professor in the Department of Psychology and Program in Neuroscience at Florida State University. I've been interested in studying the functional organization of the gustatory system in rodent models, with a very heavy emphasis on behavior, and I'm also interested in the controls of ingestive behavior.
Lindsay.
All right, so that leaves me. I'm Lindsay McPherson. I'm assistant professor here at the University of Texas at San Antonio, and my lab investigates taste, connectivity, and wiring of taste circuits through the tongue and gut.
Okay, thanks everyone. So we've seen a few recurring themes over the day. The first being that tools and receptors in circuit anatomy are fundamental to understanding the specificity of coding and behavior. We've also heard about how the availability of new tools is debunking dogmas that positions the periphery as a secondary negative modulator of central pathways and behavior. And finally, we also kind of hit on this final point that the story of gut-brain communication is really the story of vagal afferent transmission. There was lots more, but these are sort of the things that I thought that we could kind of bear out each in turn. So I thought we might first spend a few minutes talking about the first part of that in terms of understanding the sensory space of the alimentary tract of the tongue versus the gut. How well do we understand the sensory coding space at both of these areas at the level of the epithelial cell? And can we think about the entire, are we thinking about the entire alimentary tract as a single distributed sensory system with the tongue and gut as sort of two poles that each impinge on central pathways to do many different things over an evolving time course, and that ultimately promote health and drive ingestive preferences? I'm kind of trying to imagine, or have you maybe think about what some of the organizing principles are here for getting at some of the sense of this as a sensory system or multiple sensory systems. Who wants to go
Wow.
first?
Okay,
Take it how you will.
I'll take a stab at one part of it, and I'll let you know sort of what my own thinking is. But I mean, it's a great question, and I think we're still in our infancy of understanding all this. But like I sort of alluded to earlier, I think in one sense these systems are segregated. So for example, I don't think that if you intube sucrose into the gut, that an animal experiences sweet taste. All right. So right off the bat, you can see that the way that these stimuli are interacting with the receptor systems and the kinds of neural events they're generating is different. But at the same time, I think that they do interact in the service of particular types of behaviors and physiology. And so, and I think, at some point we've in the taste field, we've been arguing about coding. This has been going on for decades, and there's individuals that believe in spatial codes, like which neurons are active is the code for taste quality, and there's others that believe there's a temporal component to that. And there's some that believe it's a little bit in between. And then in the spatial coding models, you've got the labeled line type of individuals and the across neuron pattern individuals. And again, as I alluded to before, I think in the end it's going to be important to understand, at least for the people studying taste, that taste quality is only one dimension that is coded, and there are other features of the stimuli that are important. And like you could have stimuli that turn on salivation, right? And there might be other neural channels that are signaling that, and they may be broadly responsive to respond to like bitter and sour stimuli. And so I think ultimately we're going to have to come to grips with that. And I think the same thing is going to be true for understanding vagal afferents. It's a little trickier, I think, just experimentally, but we've heard some wonderfully, beautiful talks and elegant procedures, Vaughn and Diego, and so I think that it's possible, and it will be important to understand that. That's my own opinion.
So is there a one-to-one? Go ahead.
Oh, sorry. So just to pick up on something I think you mentioned at one point in your talk, Alan, was the ability for the mouse to detect and to figure out what it's tasting only requires a few taste buds. Did you kind of mention that at some point?
Yeah, well, okay, so I didn't really, I don't want to get nailed down on the number, but you don't need the full, at least behaviorally, because we see what happens as taste nerves regenerate. And when they regenerate, function returns as measured in a variety of our tasks. And incidentally, regeneration is a very important issue in some of these nerve transection experiments. But we can count the number of taste buds that come back, and rarely do they all come back to the complete normal complement. There's always a decrease. Yet the animals seem to be doing okay, and you can sort of track how many taste buds an animal has with respect to performance, and it's almost as if that there's sort of a certain minimum number, and sort of you get over that, then the animal has enough information to do the trick. However, the caveat, of course, is that you could be having neural reorganization going on centrally, an upregulation of these certain aspects of the circuit that might be helping the animal get the job done with less taste buds. So that's sort of what I meant when I said that.
No, and I think it's interesting too, because like there's a lot, I think, of in every system there's redundancy, right, to make sure that you can get these important signals that you need to inform yourself to avoid these toxins or to identify good food sources. And so it kind of makes sense to me that you wouldn't need all your taste buds to be functioning at 100% to actually do behavior. But then at the same time, this makes it difficult, right? When you're perturbing the system with all of this redundancy kind of in mind, like you're talking about these issues with, looking at these taste modifications, and sometimes, it has really no that much effect on the behavior of the animal, but yet there may be something happening within the peripheral taste system, but at some point, like how does that translate to behavior? And it's a tricky problem, and I applaud you for doing these very careful psychophysical assays to tease this out.
Yeah, I mean, thank you. And honestly, I just, a lot of times, and I think this is true for all of us in our work. You have a little bit of frustration, like when you got disparate data from different groups, and you're trying to make heads and tails and figure out what's going on. And I think ultimately the solution to that is just to keep collecting data and getting information and using different approaches and different behaviors. Because you may make a manipulation, the peripheral gustatory system has no effect on one behavior, and we have done this, but it has a big effect on another behavior. And so, you want to bring to bear as many different types of behavioral approaches. And I think what we're all trying to do is search for converging lines of evidence to support, a particular model or, view of the system.
Yeah, coming back to the original question, I agree with Alan. There are many similarities between the oral and the gastrointestinal, but many important differences. And one very interesting aspect that we don't quite understand is that, unlike taste, gut sensing is mostly unconscious. It's not a process that is accessible by whatever neural circuits are responsible for awareness, right? So as Alan put it, the animals won't experience sweetness if you put sugar in the gut, even though there is a very obvious behavior effect even when you bypass the mouth. So why is it that we are so clearly aware of sweetness and how good it is, and it is in the mouth, and which part of the mouth it is, and why we are so blind in terms of consciousness in regards to where the stimulus is in the gut and what it is in the first place? Is it because we have much more behavioral control over our craniofacial system, over the gastrointestinal system that we can't really control consciously? But there is some fundamental difference as the tract goes down, and the central circuitry underlying taste perception and gut sensing must differ in a fundamental way, in terms of why, say, the prefrontal cortex or other areas are much less interested in gut inputs. And I think this is important for understanding how food intake habits are formed and overeating and so on, eating disorders.
Ivan, it's the way people are thinking about this in terms of forming associations in some, even though it's proximately so distal from the ingestion point for the animal, because it seems like to be a teaching signal, there has to be that temporal connection.
There is obviously an association, right? But because we have conscious access to, say, taste sensation, we tend to attribute the outcome of the association to taste. So we may like caloric foods because we may say we like them because they taste good or they have a pleasant aroma, but part of this liking may be unconsciously driven by unconscious cues arising from the gut. And we don't understand that. Dana Small, Mark Schatzker, and I wrote a review some time ago about this duality in the feeding system, right? This is split between conscious versus unconscious perception. and the extent to which this actually matters. But clearly, we don't have the same type of sensations in the gut compared to the taste system. We are much less aware of what is going on and why is it we don't understand. Even though these unconscious processes clearly can change behavior in a sometimes dramatic way.
Could I qualify that just a little bit? I agree completely, but I wouldn't, I'm not willing to go as far as to say that all taste input is conscious, all right?
No,
so
right.
I think, you know, because I think this hedonic domain and this physiological domain, you know, it can be driven by signals from the gustatory system, but doesn't necessarily have to be, you know, brought to like conscious awareness. So I think that's just one,
I don't think,
qualification.
what is clear is that with taste you can obtain subjective reports, sometimes extremely accurate from subjects, in a way that is essentially impossible when stimuli are administered to the gut. So
and that's, yeah. And I think, like Selma said, I really believe this is true, that given, you know, the taste system's right at the opening of the tube, and especially for rats, they can't vomit, you know, and so once they swallow it, it's in there, and you know, so they have to make these decisions. Now, you could say that a lot of it, why do you even need to have conscious perception? But I think it really is so you can learn about the features of the stimulus and pair that with these post-ingestive events, whether they be positive or negative, to help drive the proper behavior. And so I think that's at least one part of it.
Yeah, I think Lindsay Shear's work points a lot to that, about how these vagal responses then alter the taste system at the very opening. So, I mean, that loop is really important. I'm done with that.
I do have one thing to say on that. I don't, is it necessarily that the vagal sensing is subconscious or is not a conscious precept, or is it simply that we don't have the language to describe what we are experiencing? So there's been a lot of studies about different cultures that have different languages are better able to describe what their sensory world is like. So it could simply be that we haven't figured out the right words to talk about like what a satisfying meal is. But we all certainly know, like, a good meal, you feel it in your gut. We have these colloquial terms, gut feelings, because there's something there. We just don't quite have the words to talk about it yet.
Yeah. I mean, I agree. And I think somewhat, obviously I focus more up at the brain, but somewhat is because there's a lot of things going on at once in the gut, right? You've got, in addition to sensing the glucose, you've got the mechanosensing going on. You've got a lot of different things that all act in consort at the same time. So, and I understand that there's also complexity in taste, but I think that the GI is just that one level up more of complexity. And it may be that you get some integration of those senses in terms of stimulating the vagus up into the brain, and it's whether two things are there at once will make a difference, not necessarily one alone.
I have a question for my colleagues about the innervation of the gut. You know, so much effort now is focused on the vagus for good reason. We saw today why. But, you know, I wonder how much information and what its role is through the splanchnic afferents, and, like, you know, what role do they play? I mean, I know it's been hypothesized, well, more than hypothesized, that it's very important with pain. But, you know, I'm just curious what you guys think. I mean, do you think that there's critical information going through the spinal cord route?
Yeah, I mean, I totally agree. It seems to us that the spinal system is absolutely fundamental for processes that are not nociceptive. I think again, it's a technical challenge. It's very hard to access this ganglia. First, there are many, at least the nodos, there are only 2 of them, right? But the DRGs are are all over, and sometimes, especially like the thoracic level, they are very difficult to access, to manipulate, and get the animals to recover and do real behaviors. So again, the importance is underappreciated for because of the technical difficulties of accessing the ganglia, in a similar way that happened to the nodules that now is becoming more mainstream. People have been able to do the surgery, and there is this vagus mania right now. But the DRGs, yeah, I believe they are very, very important, and we have to really get to share techniques and approaches so that several labs could actually work together to really figure them out, because they are, as a problem, a more complicated problem than the vagus due to the sheer number of neurons involved.
Yeah.
And the fact that is not a direct input to the brain, right? Then you have the dorsal horn aspect, and you get top-down inputs and so on.
Yeah, and I definitely have to echo what Yvonne was saying as well. And so some of the tracing experiments that we've done, at least monosynaptic tracing, from these sensory epithelial cells, there are a lot of DRGs that get labeled. And not all DRGs sense pain. It's just that that's a very real stimulus to us. And as you move down the digestive tract, right, there's more vagal innervation at the proximal duodenum. There's more DRG innervation at the distal colon. So I'm sure the two systems are working together.
And similarly, we're going back to the tongue, you know, that the trigeminals that are all innervating on the tongue. And so you get all these somatosensory inputs. You know, you and I, Maya, were both trigeminal type people in our past lives. And so I think maybe, you know, it is hard to access the DRGs. They're all encased in, you know, the bone, but the trigeminal is a little bit easier. Well, you know, if you're able to suck out the brain, it's kind of nasty, but you can see them.
The brain.
I know I'm a terrible neuroscientist. I just remember the brain. I look at everything else. But yeah, but these things, essentially the same things are happening too, like kind of, you know, tying back in with the taste system. The trigeminals are right there. They're you know, surrounding the taste buds. You get all the somatosensory input, and it's going in the same places too. So like all of this convergence, you know, into the NTS, I think is fascinating. And I think there's already been some studies about, you know, how the somatosensory system can work together with the taste system to improve, you know, the lick number and appetitive behavior and that kind of thing. So I think it's definitely going in that direction. I mean, we have to look at all of these routes of innervation and determine what's happening. But yeah, I agree. It's the technical difficulty is where, you know, where we're at right now. But every time we see this, people are making new breakthroughs on that front.
Well, it's go ahead. Was that somebody else? Sorry. So it's especially beguiling, this idea that I think Ivan, Diego brought it up, I guess in relation to work that Maya, you did with him, this idea that the periphery is now an access point to the nervous system by a viral pathogen insult, basically, and that this can be, I mean, it can be understood as like a new focal point for not only motivation type stuff, but also for degenerative type processes that can access through the lumen. That's incredibly scary and but super exciting. And you know, there's work on Parkinson's. It's already sort of headed in this direction. How much of that is sort of I mean, do we see degeneration at the level of the nodos, for example, or the vagus? Like, how do we imagine that would sort of happen and affect the brain at multiple synaptic levels? Do you guys have any thoughts about the next steps in studying this part of this?
Yeah, I mean, what is clear is what is known is that first of all, the enteric neurons seem to be deeply affected in some diseases like Parkinson's and maybe Lewy body dementia. And one of the ideas that has been dominating this field of the gut-brain role in Parkinson's, is that the parasympathetic neurons innervating the gut from the dorsomotor vagus are some sort of retrograde conduit for alpha-synuclein to travel from the gut to the brain. That has been the line of thought of many investigators, and this is a strong topic now, like the peripheral spread of alpha-synuclein to cause Parkinson's. But I mean, there is a lot of controversy too. Some people believe that the disease may actually start in the brain and travel to the periphery. Some others think there are two subtypes of Parkinson's, one peripherally generated, another one centrally generated, and so on. But in terms of pathology, the damage to enteric neurons and the dorsomotor nucleus of the vagus is well established from autopsies of patients who passed away. This has been documented over several years now, and this hypothesis has been known as the BRAC hypothesis, after the name of the pathologist who observed this pattern initially. Is certainly consistent with this connection between the gut and the dopamine system, because the dopamine neurons are the main target of degeneration in Parkinson's. So,
but at least two synapses away, right? At least
yeah,
two.
but what we know is that the alpha-synuclein goes through the brainstem, and the early signs of Parkinson's before motor problems like sleep disorders, REM sleep disorders, constipation, and other issues that arise early in the disease, they seem to be caused by by alpha-synuclein reaching the lower brainstem in a relatively intermediate state before it actually goes onto dopamine neurons and lead people to look for a diagnosis.
So I'm actually wondering about this idea of what the coding space is for these epithelial cells at both ends. Are we assuming that there's always going to be a one-to-one mapping between what's appetitive, what we imagine is appetitive at the tongue, and what's appetitive or aversive at the gut? Are these intuitions that we are using, or I mean, there is real data, and there are, I guess, well-known and well-characterized responses that are particularly aversive, and sucrose, of course, is appetitive. But are we, I mean, are we imagining that they're always going to sort of be anchored from at one point to the other?
I would say that as food goes through your digestive tract, right, stuff breaks down, and so at different parts of your GI tract, there are going to be different signals that are being sensed. And so sucrose, for instance, in your colon is not exactly a good sign, but sucrose at your tongue is delicious. So I wouldn't be surprised if we see that there's a change in how things are perceived, if I understood your question. That correct?
Yeah, to follow up on Maya's point, adult rats are basically lactose intolerant, and there's been, in fact, Lindsay did a nice study, Lindsay Shear did a nice study looking at conditioned avoidance versus conditioned aversion, measuring how much animals drank and whether they were also exhibiting this taste reactivity. And animals initially seem to like lactose, and then after several pairings they start decreasing their ingestion of it and avoiding it. But if you measure their taste reactivity, it's still pretty ingestive. And so the point is that when you're looking at at least intake, lactose is at first promoting intake, but then because of its consequences in the gut, it's the animals learn to avoid it. It's not quite what you're asking, because I think, I don't know if that's so much coding as much as just kind of the consequences of the ingestion changing the nature of the stimulus, but that's one example that comes to mind.
Well, it seems like a daunting task to have to map the entire length of the alimentary tract. So, I mean, I assume that there are certain points at which, based on the anatomy of the vagus, that are particularly sensitive to guiding behavior, presumably. I mean, is that true?
Yeah, well, I'm just glad I work in the taste system. That's hard enough, So the rest of the tube, that's somebody else's problem.
What are the next big steps in establishing? What are sort of the big outlying questions now for this gut-brain axis? Maya, maybe you can speak to some of that.
I mean, so this concept of sensory transduction was like a fast communication between the gut and the brain is this fascinating new field. And we've already kind of touched on this, which is, what is it exactly doing? Like, why do we need fast signaling in the gut? And how is this affecting our behavior? And so kind of pulling a couple of different points together, I think that actually your point of how the gut is being mapped onto behavior is certainly a great question. So what is it that the colon is signaling? So the colon has all of this microbiome in there, and which has been linked to a lot of different emotional states and behaviors and things like that. So does that mean our colon is more like a guide to how we're feeling, and the tongue is more what we decide to put in? Or like, and then what is the middle of the gut trying to tell us about fats and proteins or something like that? So I think that that's one of the questions that I really have on my mind is what is the fast transmission? How is that guiding our behavior in different ways as the food transfers through the entire GI tract?
I think it was very interesting too in Ivan's talk, where he started off about showing the vagal stimulation as being kind of one of the last ditch efforts to improve depressive disorders. And so just like grossly stimulating the trunk of this nerve has a positive effect. And so, I mean, and he paired that with the idea that this is kind of counterintuitive with what we were thinking about originally as, the vagus nerve is like the brakes on the system. Stop eating, you're full now. So, so can you elaborate a little bit about this and how, where is this, like the dopamine link, obviously, but I mean, there's got to be different sub components to that. And how is just like roughly, stimulating this entire nerve a way to kind of improve mood?
You're asking me? Okay.
Yes.
Yeah. So yeah, that's a great question. I don't think people know it. It's kind of so when you stimulate the nerve as a whole, it works, but you can get a number of side effects and problems, and including potentially dangerous cardio respiratory effects. So we don't quite understand where the positive change comes from, or it could be that it's not even sensory. It could be a parasympathetic effect on the body in general that is reflected as a relative decrease in sympathetic stressful type of signaling. So this type of for improving the theory and maximizing the number of people who may benefit from it, it is very important to know first which population of vagal neurons are underlie the positive effect of the stimulation, and in addition, whether there is a particular organ that is innervated by the vagus that is the origin. For example, it could be the gut, as the gut seems to be according to our mapping, the only part of the body that is connected to the reward system via the vagus, for example. But there are no tools for this type of specific targeting in humans yet. So it's a very important question from a even clinical point of view, and it just requires work on separating organs and sensory versus parasympathetic branches, and the testing in experimental models of depression, which are the effects of each of those components, to see whether the overall effect is what accounts for the overall effect. But I believe that in real life stimulation in patients, you get more than you need for mood improvement just because you are stimulating the whole nerve.
Yeah, I think it's really cool because of Suzanne's work also looking at, just direct stimulation of these vagal terminals. And so, I mean, I am still very much, a novice at understanding these circuits within the NTS. So your talk was super informative to me. Thank you very much. And no, but it is fascinating how, all of these signals can be modified. Can you talk a little bit more about that and in relation potentially to therapeutic target?
Well, because I think there's a couple of different things, and I'd like other people's opinion on this, because one thing that we do see when we record, so there's a lot of different populations in the NTS, and there's a lot of different types of inputs that are coming in. But other people as well have seen this, but when we record from specific populations, it's not always a one-to-one. So when you stimulate the incoming vagus, you don't just get one excitatory EPSP. I show the simple one because it's easier in a talk, but often it's a compound EPSP which might have two different separate inputs coming in. So there is some convergence at the level of the NTS. And in addition to the direct connections, you also have a lot of polysynaptic connections. So stimulating the vagus activates another NTS neurons that then feeds on to the neuron you're recording from, which gives you another level of integration of different information that is coming in. And I'm always curious about what that is, because different populations, so for example, the catecholamine gets a lot of direct connections. The leptin receptor expressing neurons get a lot of polysynaptic inputs. We don't know the consequences or meaning of that. The implication is it can sense from more different types of inputs coming in, but we don't know what the implication of that would be. And I do find it fascinating whether you can mimic vagal activation to drive it, but even within those populations of NTS neurons, so for example, there's been recent work on the CCK neurons. One of their projections to the parabrachial, and often the parabrachial, so there's one parabrachial circuit, which Ivan talked about, that can lead to positive affect. But there's also another projection to the parabrachial nucleus that leads to CGRP neurons that can lead to a negative affect as well. And so it's interesting how exactly those circuits are interconnected. And it comes back to the question someone asked about which neurons may mediate the positive effects on dopamine that Ivan's seen. It's like, that's a really interesting question to me. Which neurons are actually the ones doing it? It's likely that it's more than one type. It could be just only one, but you would imagine stimulating the whole vagus, you're going to be recruiting different populations that are in the NTS. And I think understanding more about the type of information that they're getting, how they integrate it, and how that contributes to what's being sent on is an important question. I don't know if that answered the question you were asking, but I am curious to know what other people's opinion is on how much you're going to get integration of different vagal activation, so the different pathways, how much they're going to talk to each other. And when you were talking about the spinal, you can get integration of lots of different types of information coming in. The taste obviously comes into rostral NTS, and Susan Travers has spent time looking at rostral and caudal NTS. So there's a lot of different ways that things can be impacted. It's a complex problem, right? It's what keeps us in keeps us going, right?
Suzanne, before you open up for people's opinion, I was wondering, because you talked a lot about how much heterogeneity there is in the NTS. Is every NTS neuron unique? Like, if you break it down to neuron by neuron, would you then find that each one is solely unique? And so if you were to ablate one, you would have like a specific deficiency.
I think that's a really, really interesting question. It probably isn't that each individual one is, but when you think about, so they're broken down by multiple things. They express different transmitters. They project, even if they all express the same transmitter, one projects here, one projects here. Even if they project to the same place, then you're talking about what information they're receiving. Is it CCK? Is it serotonin? So in theory, you're getting more and more uniqueness with each neuron, right? The more levels you get, because then you can add in, okay, so does that neuron now respond to MPY inputs coming in or acetylcholine inputs coming in? I don't think it's probably down to one. Just that seems a little harder for me to see, but yes, I think you've got a lot of things that are all acting in concert. And whenever we do any manipulations, that's always a question, right? Because often it depends on what we use to drive it. Even if you're just looking at catecholamine, if you use dopamine beta hydroxylase to drive it, that's different to using tyrosine hydroxylase. Not just because the promoters themselves may have different expressions, but it depends also on how you drive it. And to the effects that you might get. And that comes to vagal stimulation too. I often think of NTS neurons as Goldilocks neurons. It's like they want some stimulation. And I think we all know that when we're completely hungry, our gut's not happy when we have no food in it. And then there is, I think, association. I've never thought of it always as a negative feedback. Obviously, a Thanksgiving meal situation, you're getting into more aversive. But normally a meal, as you say, is a pleasant experience. You do get that signal to stop eating, but there's definitely a positive affect associated with it as well. So I don't know if that, but then obviously when the gut goes completely mad, that is also not a pleasant feeling either. So lots and lots of stimulation is not always a good thing as well.
Yeah. So then my follow-up would be like, how plastic is it then? Because I mean, it seems like the NTS is kind of this big, like this key zone. It's integrating a lot of things. And so I'm thinking of like childhood disorders and things like that. Does that shift your NTS one way or another? And how are you able to get it back?
Well, I agree with you. I think, how are you able to get it back? Although what is normal, that's a whole nother philosophical question, right? But yes, I mean, the NTS does seem to be extremely plastic. A lot of groups have looked at the effect of diet and shown that diet can have effects on it. The time of day seems to have effects on it. And clearly estrogen does have effects. That's why we do look at both males and females, because it's always at the back of my mind that you can have some differences. Age could have a difference. And then, as I mentioned, changes in glucose level can have plastic changes, as can fasting. I mean, the hypothalamus is also plastic in that way. It also, you get changes, and whether you consider those plastic or you're just getting activation of different pathways with different stimuli. But I think the way I look at it is it's an integrator of all the information it is receiving at that moment. And that will impact the outcome. But I think that's true for the gut too, right? I mean, its output is how much it activates the vagus is going to be a result of what is it seeing at that moment. See, I asked the question out. So, I mean, how do people see it? Do they see it as, when you're thinking about glucose activating it out in the periphery, do you think just that one pathway is being preserved all the way back up to forebrain neurons, or do you see it being an integrated response more?
Yeah, I think that's a great question, and it kind of gets back to one of my questions in response to one of the earlier comments, is like, what are some of the key things that we are left to sort out? And one is do we have a similar kind of labeled line system where signal coming into the gut about glucose is basically transmitted high fidelity this way to the rest of the brain, or is there, is it sort of more of a positive affect signal? Can these neurons discriminate the fat from the glucose in that sense? We have behavioral evidence that they can, but it's been sort of difficult to really find how those signals are encoded, and it's certainly different from the taste system in that way.
So do we believe that there'll be labeled lines in the gut? No intuitions about that?
I'm going to look for them, but I don't know if I'll find them.
Well, I think there might be, but I think the caveat is what the labels are, right? So it doesn't necessarily have to be like glucose, one for glucose, it could be one for like sugars, one for fats, I mean, so I would guess that there probably are label lines, but the trick will be to see, what are the labels.
That's kind of what I was getting to with the question about coding space. Like, we have these well-defined anchored taste receptors. It seems like the gut is still wide open for characterization of what sort of the canonical parts are that are going to be looked at. But I guess Lindsay McPherson, you have some ideas about how you want to go at it. Yeah.
Well, yeah. I mean, I think there's some nice parallels, right, between the taste system and the gut chemosensory system, but it's still, but it's a lot harder to study these gut chemosensors because they're very distributed. They're not like a taste bud where you have a bunch of the cells all together to study all at once. They're, one is here, one's there, and they're all over the place, so it makes it very difficult to study. And so that's why, these organoid systems have been really helpful, especially for Maya's work and others, to kind of like pull them out of their context and then look at what, these contacts outside of the body. But if you do want to look at them in situ, it's really tricky. And so you have to use some new tools to do that. So hopefully, GRASP will help in this. But I do hope, or at least, my hypothesis is that there's probably some subset of enterochromaffin cells, maybe the type that they're different than the typical nutrient sensing neuropod cells that are more, they detect kind of toxins and nasty things, and maybe those would be, those are the ones that they create the serotonin, and maybe that's a different signal than the ones that Maya has described, where they're much more sensitive to glucose. They are glutamate releasing, and that in itself is kind of an indication that something different is going on between these two populations. So that's my hypothesis. We'll see.
And yeah, so to kind of follow up on that, I actually have a question for Alan. In the bypass surgery, I find that fascinating that you're seeing a very similar phenotype that we see when we silence these cells. But what happens to those nodose neurons that were innervating that portion of the gut that you've now completely cut off? Have you looked at it? Like, do you have like phantom gut pains, or
Yeah. So I have no idea. We haven't looked at that. I'm not sure. The only person I know that's sort of looked at, in at least in animal models, sort of the role of the vagus with gastric bypass is Hans-Rudi Berthoud. And my apologies if I'm missing others. And when he does vagotomies, the effect of the surgery is to attenuate the degree of weight loss and intake reduction, but you still see it, and that's sort of been a common theme, that there's this sort of these redundant signals. In terms of the pain, again, I don't know how that relates to the nodose, say, versus the splanchnic. I do think, you know, there's this phenomena in humans that patients with bypass, many of them experience something called dumping syndrome, and especially early after the surgery. And, you know, this is in part due to the osmotic load placed on the lower gut. And, you know, it could be that, you know, rats are perhaps experiencing sensations like that. But I really do believe it's something more akin to a low, well, obviously lower GI because we've avoided the upper GI, but the sensations, I don't think it's something akin to nausea, because those kinds of sensations, at least in humans, usually lead to conditioned taste aversions, and we're not seeing that. So I think it's something more akin to conditioned taste avoidance, sort of like you would feel if you had cramps or something. It's not like you would completely change your palatability response to whatever that food was that you ate before you had the cramps. I know I didn't answer your question, but that's the best I can do.
Well, this is, oh, go ahead. Well, I was going to say kind of along those lines, I was wondering, so Yvonne, if you were to do, this is my kind of goes back to what I would like to somehow see done in my lifetime, is the creation of what you could think of as like an intestinal homunculus, and how it is the different behaviors like map onto the different parts of the gut. And so I'm wondering, Yvonne, if you do the same experiment that you did with just the straight channel rhodopsin vagal innervation of the upper intestine, but then you go to, like, say, the ileum, and you then stimulate the nodos, or you go to the colon, do you think that you will see the same kind of reward association?
Yeah, that is a great question that we have been thinking about. So one issue, of course, notwithstanding the potential differences, like the number of neurons that may be stimulated may be smaller, etc., but probably the meaning for the brain of stimulating the lower gut is going to be very different compared to the upper gut, right? So nutrients in the upper gut are an obvious reward signal that should be used as a teaching mechanism for developing new behaviors, whereas the presence of nutrients in the lower gut from the terminal ileum down represents malabsorption and should produce a totally different response. So I expect the type of behavioral sensations to be very different. There are some complicators, like, for example, the role of microbiome. There are many claims that the microbiome metabolites are sensed by vagal terminals, and this may influence mood, reward, decision-making, and so on. That's very likely. But I would expect that the meaning of the upper gut to the animal, it's going to be very different. And consistent with that, like the flavor nutrient conditioning literature shows that if you infuse, for example, sugars in the ileum or colon, you never induce learned preferences, right? So at most, you can do it by infusing sugar in the upper jejunum, for example.
Yeah.
Whereas if you do it in the lower gut, the same amount of sugar, etc., it's not going to cause reward. If anything, maybe aversion. And you have the ileal break phenomenon, where people feel nauseous and bloated when you administer nutrients directly to the ileum. So I expect to be a very different. Whether these different sensations depend on the vagus and to which extent, I am not sure. But I think the topography of the connections between lower and upper gut is going to differ significantly in terms of where they get into the brain.
And
right, but you're not necessarily going to be sensing nutrients, right? So it would be a
Not
different type.
yes.
Yeah.
Yeah, exactly. So yes, exactly. So we don't know what they are sensing, right? And that's the point. So what I mean is I can't think of any stimulus that, when present in the lower gut, should be used by the brain as a teaching signal. At least this type of stimulus hasn't been discovered so far. Everything in the lower gut doesn't end very well, like bioacids, food, mechanosensation. All those things produce avoidance responses to some extent. But yeah, we don't know. The truth is, these very old questions remain open, fortunately or not. But,
I'm imagining one of these 19th century phrenology maps, but of the gut. Because it's really about mapping behavior ultimately, right? Preference behavior.
well, the one behavior we haven't really talked about that much as well, in addition to the reward and satiation, I mean, there's also the work with Lisa Buechler and Zach Knight showing that, you know, these signals are also important for silencing those AGRP neurons. So giving more of a signal, termination signal that way.
Excellent. Well, this is the part of the evening where we should all be raising margaritas, but unfortunately, you'll have to do that on your own, unless Lindsay wants to organize a happy hour. But this has really been a great day. And Lindsay, I want to give you the final sign off to anyone who still may remain listening, to thank everyone. And go ahead.
Okay, well, thanks you all for coming and listening to today's talks. I hope you were as informed and inspired by all of the work that was presented today as I was. So thanks to all of our speakers. Thanks very much to Selma and to Charlie for
Stuart
for putting together this very first run of a virtual symposium. I think it went okay. So we're always learning things, but overall, I think we had a good representation here. Thanks to all the audience members out there. I can't see you, but hopefully a few of you have stuck around all day to hear this really fun discussion and panel. I thought this last hour was really informative and inspiring. Also to think of where we need to go next and how do we overcome some of these hurdles. So I don't want to delay you too long, but we'll end it here and saying thank you to all of you. And I hope we will be able to meet in person sometime soon. Thanks all.
Yeah. Thank you for organizing.
Thank you
very much. Yeah, thank you for bearing with us. This was a real treat. Appreciate it. And if you guys want, I can take it private too, if you guys want to have any last minute business. Thank you, everyone, attendees, for coming, and I'm going to take us offline.